session. And we will start with the first talk by Professor Aaron Dinner from University of Chicago, who is going to talk about estimating commuters and relative currents through dynamic approximation. So, Professor Dinner, you have 30 minutes for talk and 5 minutes for question answer. So you can start your talk now.
Great. And so you're seeing the slides as presenter mode?
Yes.
Everything looks okay?
Yes.
Okay, great. So let me begin by thanking the organizers. So far, it's been a really great meeting, and I'm only sorry that we're not actually meeting in person. So what I want to talk to you about today are some data-driven approaches that we've developed for computing kinetic statistics. And this is work from a number of my former and current students, and it's a long-standing part of a long-standing collaboration with Jonathan Ware and his student Rob Weber at New York University. Okay, so, oops, it won't let me move. Okay, so kinetics are important, and just to give you one example, a system that I'm going to show you some results for later, there's tremendous interest in being able to design insulin therapies with particular pharmacokinetics. So you might want a fast-acting insulin that you could take right before a meal, or that you could put in a pump that somebody would wear and would be continuously monitoring the glucose levels in the blood. You might want a slow-acting insulin so that you only have to take one shot during a day. So insulin, the protein hormone, it takes a variety of oligomeric forms. When it's in vesicles in the pancreas and then secreted, or in a therapeutic formulation, it's typically in a hexameric form. It then dissociates to make dimeric form. That's the predominant form in the blood. And then, as Biman Bagchi mentioned, what was my morning, I guess, yesterday evening, for some of you, then this dimer has to dissociate to a monomer in order to bind to the cell receptor. Okay, so you can see that actually the different transitions between oligomeric forms have different kinetics. And so we would like to be able to understand those kinetics and the mechanisms underlying them, because each of them is a point where one could actually manipulate the system to try to get better overall kinetics, okay? So that's just a motivating example. I'm sure many of you can think of your own examples of interest. So I don't need to tell this audience that because there's a separation in timescales between the time step of a numerical integrator and events of interest, right? In insulin, you saw that that graph had an axis in hours. Because of that separation in timescales, you can't simulate these dynamics directly, and you have to do something else if you want to get at the kinetics. And we've heard about a variety of different enhanced sampling methods already in the meeting. The one that I want to talk about briefly, because it's going to set the stage for what I'm going to tell you about, are Markov state models. So the idea is that you have some dynamics, however generated. We'll talk a little bit about that in a bit. And then you defined states. You bin the trajectories into those states, and then you determine the state-to-state transition probabilities. Once you have that transition matrix, then in principle, at least at the coarse-grained resolution, you can calculate the full evolution of the system, and so any quantity that you might want. Okay. In practice, what is often done is simply to look at the eigenvalues of the transition matrix, because those provide information about the slowest relaxation timescales. But there's an issue with that, which is that the process of interest might not be actually the slowest one. Maybe I'm interested in how, say, a hexamer comes apart into a dimer, but actually the slowest relaxation timescales are the unfolding of the monomer, for example. An additional issue is if I don't specify a reaction a priori, then actually the computation of the statistics that I would like, so things like committers, mean first passage times, rates, those will actually be biased by using the transition matrix that I would construct just from all of the states without knowledge of the reactant and product states. So what I want to tell you about is an approach that we've been developing the last couple of years called the dynamical Galerkin approximation. And what it does is it shifts the focus to actually directly solving for kinetic statistics. The way that it does that is cast those statistics as solutions of an operator equation, where that operator is a so-called transition operator, which tells us about the expectation, the evolution of the expectation of functions. So in some sense, the way you could think of this dynamical Galerkin approximation, or DGA, is as a generalization of Markov state models, but one where we've actually specified what is the reaction or process that we're interested in. And then we're going to be able to correctly account for the, in essence, reactant and product states when we construct the appropriate transition matrices and then solve these operator equations. Okay, so that's the basic idea. So to make this a little bit more concrete, one very important statistic that we will want is the committer. So Ron Elber introduced this statistic yesterday. It's the probability that I go to the product state before returning to the reactant state. So committer of close to 0 would be part of the reactant state. Committer of close to 1 would be part of the product state. And then a committer of 1/2 would be a transition state. I have an equal likelihood of going forward to the product or back to the reactant. So in some sense, this committer is a perfect reaction coordinate. It's always telling us about what's the likelihood that we complete a stochastic reaction. Moreover, I'll show you that we can actually compute other statistics from it, like rates. But traditionally, this has been a very hard statistic to obtain, because traditionally what people have done is they've started with a configuration and then randomized momenta and then run new molecular dynamics simulations. And so then to evaluate the committer for a single configuration actually takes many more simulations. Okay. So what we're going to do instead is solve an operator equation for the committer. And that operator equation is T, Q equals to T. Ron actually had a version of this equation in his talk, just using different symbols. And then we have boundary conditions associated with that equation. There's 0 when we're in the reactant state and 1 when we're in the product state. Okay. I could write down similar kinds of equations for other kinetic statistics that I might want. The so-called backward committer, so that's the probability that I last came from the reactant, and actually the stationary distribution, so the potential of mean force in effect. If we have those three statistics, then we can actually compute the flux from the reactant to the product. And moreover, we can break that flux, which is tantamount to the rate, down into currents through different regions of space. And those tell us about what are the pathways that are contributing to the reactant. So these are the kinds of statistics and the kinds of equations that we want to solve. Okay, so the way we're going to solve those equations is that we're going to expand each statistic in a linear combination of basis functions. So we're going to have one that's going to account for the boundary conditions. And then we'll have a sum over ones that solve the homogeneous problem. That way we can add and subtract these phi functions without going out of the space. Okay, and then what we do is we proceed very similarly to what we would do if we were setting up the secular equation in a quantum mechanical problem. We take this basis expansion, we substitute it into the operator equation, we multiply by another basis function, and then we integrate to form inner products. And if we do that, we convert this operator equation into a linear algebra problem, just like quantum mechanics again. Okay, but here everything, of course, is classical stochastic. So these matrices C, they have elements that are of this form, phi, t, phi. And the issue is that in contrast to quantum mechanics, we don't know actually the form of T explicitly. If this was a simple diffusion, we could write down a differential operator. But actually here we have a finite time, and everything that is happening within our molecular dynamics simulation is going into that. And then we typically don't want to work in the full configuration space. We usually want to work in a somewhat reduced space. And so we have actually a kind of coarse graining where we don't know actually those dynamics a priori. Okay, so it might seem like that's a problem, but actually that's where the data comes in. So what we can do is we can actually estimate these matrix elements from molecular dynamics data. So we generate a database of trajectories, and then we pick up time points that are separated by a time tau, the lag time. And then we're just going to approximate the action of T on phi j by this difference of phi j at tau from minus phi j at 0. Then we just multiply by phi I, that's this part, and then we sum or we average, and that's the bracket. Okay, so basically from the statistics of trajectories, we can estimate these matrix elements. We can then solve the linear algebra problem, get the coefficients, and reconstruct the kinetic function of interest, like the commit. Now, something I didn't emphasize before is that when we form these inner products, it's an integral over a distribution, sampling distribution mu, and that can actually be an arbitrary distribution. So that's going to give us a lot of freedom to optimize the procedure. So it can be an enhanced sampling approach, for example, trajectory stratification. So that would encompass things like the milestoning that you saw yesterday, or non-equilibrium umbrella sampling, which is an approach that we've developed in parallel to Ron over the years. But you're going to see that actually it could be a less structured procedure. And so we can actually build up a database with a lot of flexibility. Okay, so I'm going to show you results for two systems. The first is really to validate the method, and then the second will be an application. So The first system is the well-studied trip cage mini protein. So this is a 20 residue designed sequence that's been studied by quite a few groups, both experimentally and computationally. You can see in this in the picture on the left that there are two folding pathways, one where the hydrophobic core forms first, and then this alpha helix, and one where this alpha helix forms first, and then the core. And we're going to make use of some very nice simulations from Peter Bolhaus some years ago. And what Peter did was transition path sampling, and then actually the difficult process of shooting in order to find, I think it was 12 transition state structures. So we can compare to those, okay? Okay, so the first part of our procedure is that we have to actually generate some data. And in our case, because we knew a lot about this system, what we did was we distributed initial points in the space of Rmsd and then this 310 helix Rmsd. So those are the blue dots here superimposed on the potential mean force that I'm going to calculate in a moment. And when we did that, actually, we found that we hadn't adequately sampled the unfolding of this alpha helix. And so we actually added a second set of simulations where now we're actually distributing points in the end-to-end distance so that we can ensure that we unfold that alpha helix. And so in total, what we've got are a little more than 1,000 trajectories with each one is 30 nanoseconds. And so we have about 30 microseconds of simulation. So it's actually really not very much when you think about it, okay? I'll show you, we get really actually excellent results with it. Okay, so before we get to actual kinetics, let me show you the potential of mean force. So to apply the procedure, we have to choose a basis set. I could use a basis set of indicator functions. That would be the sort of typical Markov state model states. Here, actually, what we're doing is we're choosing pairwise distances. So we have the freedom to choose a continuous function. We choose 153 pairwise distances between selective C alphas over the sequence. And then the basis functions have to satisfy the homogeneous boundary conditions. And so we multiply by the distance to states A and states B. So that takes those functions to 0. And then we do some PCA basically to, in essence, order and orthogonalize the basis. Okay. So once we do that, we can then make, in essence, free energy estimates for all of the states in our database, 6 million states in our database in this 153-dimensional space. And then we can project it down to different pairs of order parameters or collective variables to see which ones tell us the most. So here I'm just testing. I think it's 5 different order parameters. I'm going to show you our most of the results in the Helix Rmsd and the full Rmsd, because that's what Peter used in his study. But we actually find from our calculations that we get a little bit better description of the system with the Helix Rmsd and the 310 Helix Rmsd. And so actually, if you look at our published results, we have a lot in those coordinates. Okay, so now we're going to calculate the committer. Again, we do that calculation in 153 dimensions for 6 billion structures. So that's something that I never thought we'd be able to do. And then, just for the purposes of visualization, we are projecting down to 2 dimensions and averaging, okay? And then we're comparing to Peter's results. you can see that the white regions here between the red and the blue, which correspond to committer equals a half, they correspond very well to the positions of his transition states, these red symbols. And furthermore, you can go in and you can look at the individual structures and compare them to his, and again, they look good. Okay, so we feel that that means the method is working. Then we can actually get a quantity that hasn't been computed previously, but is actually quite important in so-called transition path theory, and that's the reactive current. So this is, in essence, the local flux through a surface. And so in Peter's case, he showed just a couple pathways showing, in essence, the two pathways. We get a much more statistical view from the current. You can still see, in essence, the two pathways, but now actually from the color of these arrows, you can see that actually this one that kind of comes down and goes along the bottom is actually a much more populated pathway. And we can formalize that by dividing up this space and then integrating in order to calculate the flux through each of the two pathways. Okay, so we can make that quantitative. So overall, what the trip cage calculations are showing us is that the procedure is working. So now what I want to do is turn to insulin. I already basically introduced the problem. What it turns out is that actually in therapeutic formulations, there is often phenol included. It was first introduced as an antimicrobial, but it turns out that it actually stabilizes the hexamer, and so it increases the shelf life of therapeutic formulations. And so by understanding how phenol is released from the hexamer, we can potentially learn something about how we might make other molecules that would further stabilize the shelf life of a therapeutic formulation. Okay, so that's the motivation. Okay, just to orient you, like I said, Bowman introduced insulin in the previous session. He was looking at the dimer that you can see on the right. Here with this characteristic anti-parallel beta strand at the dimer interface. The hexamer you can think of as a trimer of dimers. So if I rotate the hexamer, I've got one dimer here, one dimer here, and one dimer here, okay? You can see that there are 6 phenols bound in this structure. And it's a little bit difficult to appreciate from this representation, but the phenol is really buried. If I were to show you all the atoms, you wouldn't be able to see the phenol at all. So it's really a rare event for it to leave. Okay, so the first thing we have to do is we have to kind of do some exploratory calculations with a view towards being able to generate the initial points that we're going to run unbiased dynamics from. And so we did that through a kind of steered molecular dynamics method called adiabatic bias molecular dynamics. It gently ratchets the phenol out of the binding pocket. The first thing that those calculations show is that actually the escape channel opens quite easily. It happens quite frequently in our simulations, and that hadn't been seen before. And then if you count the closed and the open states as separate pathways, then we see 6 pathways of escape for the phenol. So these sort of van der Waals spheres are the phenol with many configurations superimposed. And so pathway 1 or pathway 1A, which is similar but has an open channel, that's the phenol coming up through these 2 green residues, which are so-called gatekeeper residues. The phenol has to go through the gate. Pathways 4 and 4A are the phenol going down through these 2 black gatekeeper residues. Pathway 2 is the phenol coming, in essence, between the two sets of gatekeeper residues, so avoiding, in essence, the gate. And then pathway 3 is actually quite different than the other pathways that I've mentioned. It's going out through the dimer interface. So it's a different direction, in essence, going back, as shown here in yellow. Okay, and some of these pathways, I should say, were seen previously. In particular, actually, Harish Vasith, who I think is attending the meeting, did some really beautiful simulations now actually quite some years ago, where they saw actually 3 of these pathways. And we recover those, and then we see some additional ones, particularly ones associated with this opening of the escape channel. But in all of these cases, both the earlier simulations as well as the simulation that I'm showing you here, it's driven in some way, so they're biased. Okay, so what are the kind of questions that we want to ask? So just what are the unbiased dynamics? What are the relative weights of the pathways in that case? Where are the transition states? What are the rates? And then how would mutations impact the mechanism? Okay, so the first thing we have to do is we have to generate some unbiased trajectories. So we pick points out of the ABMD simulations that are roughly evenly distributed in distance from the binding pocket. We run almost 700 40 nanosecond trajectories. So we get 28 microseconds of sampling. Again, actually not that much when you think about it, so it's pretty economical. We use a basis set similar to the one that I showed you before, except now we're working in a somewhat larger space of almost 300 pairwise distances. Okay. All right, so now we can apply our method. We can compute potential mean force, reactive current, and committer. I'm going to show you each one of those in turn. I'm going to show you them projected onto counts of contacts along pathway 1 and pathway 4, because that provides a pretty good separation of 4 of our pathways, actually 1 and 1A and 4 and 4A. Okay, so just to orient you, here at about 60-30 is phenol in the binding pocket. So it's in essence the crystal structure. These couple other minima, those are with the channel open and varying degrees of the phenol coming out. And then when the phenol is totally unbound, we're at 0, 0. There are no contacts, okay? All right, so that's the potential of mean force. We have minima at key states. Then we can calculate the currents along pathway 1. In essence, what happens is we loop out in this space following these arrows. We can look at the structures at kind of key landmarks along there and kind of see what's blocking the way for the phenol, and then eventually we loop back in to 0, 0. And I should say, with channel closed, you have a somewhat larger loop than with the channel open, and that's just because you're able to make more protein contacts when things are a little bit closer in. Okay, pathway 4, instead we go up and then down in this representation, and as we come sort of along the axis here, then a kind of interesting thing is that there's some melting of one of the helices of the A chain. Okay, so those are pathways 1 and 4. We can look at where the transition states are along those pathways. Those are marked by the purple lines in this committer plot. What's actually really interesting is that the transition states are quite late in the process. So they're once the phenol has already gotten past the gatekeeper residues, which I don't think I would have guessed before doing the simulation. Okay, so that's pathways 1 and 4. We'd like to look at pathways 2 and 3. In this space, those 2 pathways overlap. And so what we want to do is split the data into a third dimension. And the third dimension that we use is actually the committer, right? We can calculate it for all of the structures in our database, and it should be a perfect reaction coordinate, and so we might as well use it. And you can see actually it does a pretty good job of splitting pathways 2 and 3, the yellow and brown here. And then if I just summarize all the pathways over here on the left, now colored with the dots colored by the committer, what we can now do is we can make a cut horizontally in this space around committer equals a half. And that'll give us a plane, and we can then divide up where the different pathways are cutting through. And then we can integrate over each of these rectangles the reactive current, where we get the populations of the different pathways. So what we can see is that pathway 4A, that's with the channel open following the pathway 4. So the black gatekeepers, that's the most populated pathway, but all of the pathways have significant representation. It's about half and half open and closed channel, which again, I think would not be obvious until you can do the quantitative calculation. And so it's really kind of intrinsically a truly multi-pathway reaction. And so what I think that says is that really actually, traditional approaches that are based on a single transition state are not going to be successful. Okay, so very quickly, let me show you some results for some mutants. So we looked at 2 mutants, B13, which is a glutamate that sticks right into the middle of the hexamer. We removed a charge to make a glutamine, so you'd expect that to affect the channel opening. And A10, an isoleucine to valine mutation, so we took away a methyl group. It's one of the gatekeeper residues, so it should affect pathway 1. Okay, so here's B13. I'm just showing you the reactive current. We can calculate all the other statistics too. And from the difference, you can see that actually pathway 4 is really actually depleted. So you can see how things shift around here. A10, I'm just showing you the committer. And here's the difference on the right. Something to notice is that actually most of this plot is very close to 0. And so that actually speaks to the reproducibility of the calculation. Just by changing one residue, most things actually don't change. Completely new data set, new analysis. Where we do have a difference is where there's sort of this blue streak here. And so what that says is that the transition state is moving a little bit later. So it's favoring the bound form, disfavoring the unbound form. So that's really all I wanted to say about insulin. I did want to make one advertisement before finishing up. Something I didn't have time to talk about is some work that we did looking at the so-called variational approach to conformational dynamics. So that would include methods like TICA or VAMP nets. And what we show actually in this upper paper is that those methods are very sensitive to the choice of lag time. And we introduce a very simple fix, which is to integrate over the lag time. And we show that actually you maintain a variational principle. And then this picture and this lower reference is what we've done is we've taken now that integrated approach, IVAC, and applied it to understand the structural ensemble of the insulin monomer in collaboration with Andrei Tokmakov, and that's allowed us then to interpret some hydrogen exchange protection factor data from Mike Weiss, which had some difficult to interpret features. So if you're using these methods like TICA or VAMPnets, VAC, then I would encourage you to check this out. Okay, so what I tried to show you is that we've introduced this dynamical Galerkin approximation. Galerkin approximation is just a basis expansion for computing kinetic statistics. You can think of it as a generalization of Markov state models, but one where we're actually saying this is the reaction that we're interested in, and we're properly accounting for the reactant and product states when we compute these statistics like the committer or the reactive current. We validated against a very well-characterized system, the trip cage, and then we've been applying it to looking at phenol release from the insulin hexamer. We've also done the dimer dissociation, although those calculations are not as well converged yet. And then what I showed you is that actually, because you're able to work in this sort of higher dimensional space, you can actually try out lots of combinations of order parameters or collective variables to evaluate how well they represent the mechanism, the committer, the potential mean force. And then we can actually get sufficiently accurate results that we can make very subtle changes to the molecule and actually see how things like the committer or the reactive current above the pathways, how those move around. So I think it's really quite a powerful technique. And I hope people are going to use it. So with that, I'll stop, and I'm happy to answer any kind of questions that people have.
Thank you, Professor Dina. That was a fascinating talk. I see there are a couple of questions in the chat box, but I think in the interest of time, we'll just allow one of them. So Jagannath, do you want to ask, or do you want me to read the question?
I can ask actually. The question actually is that what we are trying to I try to understand is that this DGA that you try to propose, and if you look at the regular maximum likelihood based MSM, and which you are trying to point out the DGA is a generalization of this MSM. I'm just trying to figure out is that, let us say, for trip cage, which is fairly a model system, is it has it been tested that the rate that you get or the timescale separation that you get from regular MSM, how different it will be that you will get from DGA?
Yeah, so at least in our hands, with our data set, we find with MSMs and DGA, we get similar results, but neither of them are actually converged. We have to go to longer lag times than we can with the 30 nanosecond trajectories. And so we're actually not at a converged result. There is a data set from the Shaw group that is like a 200 or 300 microsecond data set. The problem with that data set is that it's just unbiased, a long unbiased simulation. And so there's actually only 12 or so folding, unfolding events in it. And so it actually has less statistical power than the short trajectory data set that we generated. We have analyzed that data set as well. Yeah, so, but to answer your question, we really have to go to longer, lag times to fully answer that question.
All right, okay.
So thank you, Professor Dina. I think, yeah, you can answer the questions that are there in the chat box. So in the interest of time, we move to the next speaker. Professor Gregory Bowman from Washington University. So he's going to talk about goal-oriented adaptive sampling. So Professor Bowman, you can share your slides.
All right, sounds good. Can you see them now?
Yes, there is.
Perfect. Well, thanks so much for having me. This has been a lot of fun so far. I'm looking forward to more of it. Yeah, so I thought I'd tell you some about our work with adaptive sampling and what we've been doing with it. And thank you to all the other speakers for saving me from having to introduce Markov models. So I'll skip over this and just say that, one of the things that I've really appreciated about these approaches since, getting involved in them early in graduate school is the ability to integrate information from an arbitrarily large number of independent simulations that may not have Boltzmann distributed starting points, and to integrate these into a model that captures long timescale processes that are far beyond the reach of any individual simulation. So one of the cool things I've been involved with since the beginning is this folding at home project where we said, okay, well, if we can use arbitrarily large numbers of simulations. Sadly, the NIH won't give us arbitrarily many computers, but there's a lot of them out there. So let's go invite anyone who wants to contribute their personal computing power to help run thousands or tens of thousands of simulations of a system in parallel. And then we can take all that data on the back end and build Markov state models. And so this has been really exciting, especially in the past 3 or so years. I've taken over as the head of the project, and I guess it's more than that now, but very exciting during the pandemic. As I'll tell you later, we started getting involved in some SARS-2 research, and so we went from having somewhere around 30,000 people around the world participating in this to having well over 1 million people in the world, and really having a lot of bandwidth to try a lot of different things. But of course, this is still pretty brute force. And even with this, we're still compute limited with lots of the things that we would like to do. And we'd like to democratize, the ability to get at these complex landscapes. So we've been constantly thinking about, okay, well, how can we take these ideas further and build these Markov models with even less, raw computing time. And so we've been thinking a lot about these adaptive sampling approaches, where instead of just starting a batch of simulations and letting them go for some amount of time, and then taking all of this data at the end and building a Markov model, what we'd like to do is take a more active role in the process and iterate between running a batch of simulations, then building an MSM, and then using that MSM to decide, well, which of the states that I've seen so far should I start the next batch of simulations from in order to improve my model as efficiently as possible, without, say, redundantly sampling the same conformational states and the same deep minima over and over and over again, and redirecting that compute power to more and more interesting, maybe higher energy states. And there had been some interesting ideas on this and some cool proof of concept before I started my faculty position in 2014, but no one was really using it for applications, because it just wasn't there yet. And my thought was that, okay, well, one of the issues here is that these methods are designed to increase your convergence on the perfect map of the entire landscape, more quickly than you would get with conventional simulations where you're not doing anything. And so what we started thinking about is saying, okay, well, with lots of these systems, the conformational space is so big, we're nowhere near, getting thorough exploration of the landscape. And so let's see if we could think about how could we incorporate more information to focus our search on the parts of the landscape that we care most about. And so this got me reading about the concept of exploration and exploitation trade-offs in the optimization literature. And we came up with this algorithm where the idea is to try to amplify interesting looking fluctuations, not by modifying the Hamiltonian or adjusting the temperature or anything that would perturb the Hamiltonian or the kinetics, but just by directing where we're gathering data. So what we came up with is this goal-oriented adaptive sampling approach, as we call it, called FAST. And so the idea here is that we have some landscape, like the one I'm showing here, right? So blue is low energy, red is high energy. We imagine that, as in the case of many biomolecules, we have some point on this landscape, say a crystal structure. And we can imagine we have some progress variable in mind. That distinguishes, in a structural sense, the sorts of regions of this conformational space that we're most interested in, right? So one of the things that we've been very interested in is hunting for cryptic pockets that are absent in the structures that are currently available in the protein data bank from methods like crystallography, for example. But then as we watch these proteins fluctuate, we see these unexpected pockets spontaneously open up. And we've been very interested in the opportunities this presents for drug design, for example, or new insights into how proteins function. But you can imagine if you're going and hunting for pockets, you don't even need to know where precisely you want a pocket. You could have a progress variable like the total pocket volume in your structure, right? And so the idea would be that I would rather explore regions of this landscape over to the right than regions over to the left, given finite computing resources. And so again, taking cues from the exploration and exploitation trade-off literature, what we came up with is a process where we're going to start some simulations from our starting point, let them run for a fixed period of time in the order of tens of nanoseconds. So they're just going to explore locally. Now we're going to build a Markov model of the small swath of conformational space that we've seen. And then we're going to rank each of the states in our model based on how useful running a new simulation would be from that state. And our rank is going to have two components, right? So the first is our exploitation term, right? So this could be ranking things from smallest to largest total pocket volume, for example, or it could be from largest to smallest Rmsd to some target structure, or maximizing some distance, anything that you can measure from a single protein structure. So we'll rank things from 0 to 1, feature scaled along this progress variable. And so that's going to prefer to simulate things that are further to the right, other things being equal than things that are further to the left. But of course, we can easily imagine that there's going to be situations where this complex rugged landscape is going to give rise to dead ends. And if we just did this, we would probably get stuck pretty quickly. And so the other aspect of this is this exploration term, right? And in its simplest form, this can just say, okay, well, all other things being equal, I would rather simulate a state that I've only seen once than one that I've seen 1,000,000 times, right? Because I can do a pretty good job of guessing in a statistical sense what's likely to happen from a state that I've seen 1,000,000 times. But if I've only seen a state once, who knows? It could do anything, even if it doesn't have the structural property that I want. I could get lucky and simulation started there, wanders off and finds new structures that do. And so this is really nice now, because if we get stuck somewhere, we're going to gather a lot of data there, and that's going to get penalized, and we'll go off and try something else and see if we can reroute around whatever dead end we've hit. So just to show you this in practice, can show some simulations running in this space. So the lighter green or yellow is the active part of the simulation that's running, and dark green is what we've seen in the past. So you can see this is just random at first. We start to get some rightwards motion. We get stuck here in this dead end, but then the simulations do backtrack around this because of this exploration term. And we end up getting pretty thorough exploration of the right half of this pretty simple landscape, but with an amount of simulation that is far from sufficient to exhaustively explore this landscape. And just to contrast this with conventional simulations without any adaptive scheme, and on the bottom, what you can see is at the beginning these methods are basically indistinguishable, but as time progresses, you can see that the conventional simulations don't have anything built in that says there's a preference for stuff over here with cryptic pockets. And so they end up going in the wrong direction from our perspective, given what we're trying to do, and spend a lot of time redundantly sampling regions of conformational space that we're really not that interested in, right? So with this, we get orders of magnitude speed up, compared to, with fast compared to what we would get with non-adaptive sampling simulations. And one of the beauties of this is because we haven't messed with the Hamiltonian, we haven't messed with the temperature, we still get reasonable thermodynamics and kinetics for the part of the landscape that we've sampled. So here what I'm showing is varying the number and length of simulations for your conventional simulations on the left and fast over on the right. The color is the KL divergence between the MSM that we used for running synthetic simulations using kinetic Monte Carlo in this case, and our gold standard, the true model we use to generate these trajectories. The black lines are contours of equal amounts of data so that you can see how this tracks. And so, with numerous enough simulations, it's basically the amount of data that you have that determines how good the model is. But of course, this breaks down if the simulations are very short, right? You just can't do much starting from one point, right? But with adaptive sampling, you can see that you can actually make quite effective use of many short simulations by making this choice of where you're going to launch your simulations. And one of the important things to note here is this isn't for the whole landscape. This is showing that we converge on the right transition probabilities for the states that are along the highest flux pathways from our initial point to a set of states that are far along on our progress variable, right? So this should, in principle, do a better job of getting the whole landscape, but it particularly does a good job of focusing in on the parts of the landscape that you think you might care about. So this has been really powerful in a number of settings. I refuse to let people in my lab work on the alanine dipeptide. So we've done a number of things with beta-lactamase enzymes, trying to make sense of variants and hunting for cryptic pockets. And a third of my lab is actually experimental now. So we show that we can actually design variants of this enzyme to control its activity. And we've discovered small molecules that bind these cryptic pockets. And we were starting to branch out into other systems to show how general this is when we got interrupted by the arrival of the SARS-CoV-2 virus. And so given the generality of this platform, it was very natural to say, okay, well, let's put these other things that we're starting to delve into with myosin motors and cardiac disease and ApoE and Alzheimer's disease, and let's throw these tools at the protein components of the SARS-CoV-2 virus and see if we can help shed some light on how they work and new opportunities for combating the pandemic. So the first thing that we turned our attention to is the spike protein that's on the surface of the virus. And this is really interesting to us because the predominant structure of the spike is actually this closed up or down structure where the three subunits in the different colors here are actually closing up and burying the receptor binding motif that's responsible for engaging the ACE2 protein and initiating cell entry and infection, right? And so this is this cool conformational masking strategy where it's protecting that well-conserved interface by having it buried. But of course, like a turtle or any other predator, it has to come out of this protective state in order to hunt its prey. And because of the threefold symmetry, we imagine this like the mouth of one of these Demogorgon monsters from the television Stranger Things. And all that was known structurally about what this open state looked like from cryo-EM was this so-called up structure where one of the receptor binding domains you can see here has peaked up, this orange one, just barely exposing that key interface for engaging ACE2. But when this happens, we notice that a lot of contacts are broken. And so we imagine that there might be a lot more conformational heterogeneity to this open state, and that capturing that might shed a lot of insight into how the virus works, how it evades our immune systems and infects cells, for example, and maybe inform development of future vaccines or therapies. So we used our FAST algorithm to try to promote separation of the receptor binding domains. So this is a view of the spike now where the upper left is tethered to the surface of the virus. The lower right is where these receptor binding domains are that have to open up. And with just 10 microseconds of this fast simulation technique, we were able to capture this huge motion of this 3,600 residue system with something like 700,000 atoms in this system. So you can see this receptor binding domain has completely separated from the others over here and exposed all kinds of cryptic surfaces, including, of course, importantly, the receptor binding motif that's responsible for binding to the ACE2 receptor and initiating cell entry, but also all kinds of other surfaces that one might have assumed were buried based on all of the available cryo-EM structures. And so this is really exciting to see this, big dramatic conformational change. And so what we did was initially run this 10 microseconds of fast to capture, this big opening, and then we ran simulations started off from all over the landscape unfolding at home to get better statistics and get a robust MSM. And one of the great things is that, we could get a millisecond of data for this protein given, the million devices we had access to, but we could do way more than that actually. So we did this both with the glycosylated and unglycosylated protein. And so here I'm just showing probability distribution of the extent of RBD opening. You can see they're pretty similar. Maybe the glycans help promote opening actually, and that's something that other people have confirmed in studies that focus on that now. Going beyond this though, we also started looking at homologs of the SARS-2 spike from other coronaviruses to see if we could shed some light on what's going on that makes this particular virus so devastating. And so we looked at the original SARS-1 virus and another coronavirus called Nl63 that both also enter cells via attaching to the ACE2 receptor. And this was really interesting, right? Because what we found is that the original SARS virus here in red has the greatest propensity to adopt these open structures. Right, and so this was interesting because the original SARS virus was very, deadly and spread very well, but it didn't get that far. Right, there were only a couple thousand people who contracted the virus and were hospitalized. And so we think that one of the contributing factors is that, the spike adopted this open state quite often, and that was good for cell entry, but also a big liability because it gave our immune systems a greater chance to recognize the presence of the virus and mount an immune response. In contrast, this Nl63 virus is the most closed. And that again is interesting because it's associated with a common cold. So, it's just around, it spreads pretty readily, but it isn't a big deal. And we think that that might be because it's quite good at evading our immune systems, but less good at cell entry and transmission, for example. And the SARS-2 virus is maybe in this Goldilocks position in between, right, where it's far more closed than SARS-1. And to compensate for that, it also has some mutations in the receptor binding domain that enhance the affinity between an exposed RBD and ACE2. Right, and so it's much better at evading our immune response by being more closed, but it's pretty much equally good at binding to ACE2 in the end as the original SARS virus because of these mutations in the receptor binding domain. And so this is really cool and might give us a handle for starting to interpret emerging variants, for example. And an increasing number of works are corroborating this, right? So one study that looked at discovered that some of the antibodies that were found against SARS-1 actually bind to one of these cryptic surfaces or cryptic epitopes that we predicted from our model. And it turns out these antibodies are neutralizing against SARS-1, but not against SARS-2, even though the epitope is conserved, right? And so this is very consistent with our prediction that SARS-1 is much more open than SARS-2. And more recently, hydrogen deuterium experiments from Susan Markowski suggest that, as we had kind of hoped to see at the beginning, that all three RBDs do open up and get exposed. And so if we had even more compute power, we might be able to capture that. And this is something we've been exploring in the background. So this is a nice demo of some of what we've done, but it's just the tip of the iceberg in terms of the data we gathered. So we studied about 60 different systems, actually. So most of the proteome of SARS-2, a bunch of homologs from other coronaviruses, and some of the relevant host factors and complexes between all these proteins. And in aggregate, in the first 6 months of the pandemic, we generated about 0.1 seconds of simulation. It's a lot of data, but the Markov models and the raw simulations are all publicly available if anyone wants to make use of them. And this really highlighted another problem, which is, okay, well, if you can get a lot of data, we also need to automate not just the acquisition of that data, but also the analysis of that data, because it's just a lot to go through. And so this has been another major theme in my lab, and given interest here in machine learning and deep learning, I thought I'd show you one of the things that we've come up with, which is quite good for comparing simulations from different conditions. So this can be things like different variants of a protein, or apo versus holo simulations, or subensembles for one protein. The idea is to come up with very general scheme that doesn't make any of the assumptions of the methods we all know and love and employ quite broadly, right? So we don't have to, we wanted to relax the assumption that, big geometric changes are more important than small ones, right? Because that's not always true, or that slow stuff is always more important than fast stuff, as we heard. That doesn't necessarily apply with insulin, and certainly in many other cases. And so just to show you how we've been using this, one of the other systems we spent some time with is a part of SARS-2 called NSP16, which is a methyl transferase. So it transfers a methyl from a cofactor called SAM to the end of viral RNA to mimic the methylation that our cells perform on our own RNA. And this is another immune evasion strategy that prevents our immune systems from easily recognizing the viral RNA as foreign and triggering an immune response. And this system is really interesting because it's essential for the survival of the virus. It's also a difficult drug target, though, despite being essential, because people have come up with compounds that target the active site, and this does inhibit the viral protein. But it turns out they also have really nasty side effects, because they also inhibit the enzymes that humans have to perform the same reaction. And so a lot of people have turned away from targeting NSP16 to focus on things like the main protease and other components. But as we were reading about it, one of the things that stuck out to us as interesting is that the human homologs of this protein are active as monomers, but the viral protein is only active in the presence of another viral protein called NSP10, suggesting there might be some allostery that we could explore. And so to understand this, what we did is run our fast pockets algorithm on NSP16 as a monomer. We also did this on the complex between NSP16 and NSP10. And then we wanted to be able to compare these to make sense of what's different and see if we could start to understand what NSP10 does, and if we might be able to take advantage of that. And so, like Jagannath, we took an interest in autoencoders, which are these neural networks where you take some high-dimensional input, like a protein structure, you run it through multiple layers of a neural network, each of which is smaller, to get down to some coarse-grained or latent space. So that's the encoder. Then there's a decoder, which is a kind of mirror image of that now goes from your latent representation back to a high-dimensional representation. And during training, what you do is update the weights to minimize the difference between the input and output. And now your latent representation is this nice coarse graining of the data. So as we've heard already, this is a very powerful coarse graining method. One thing that we thought might stymie some of our efforts, though, is that, like PCA, what this is effectively doing is assuming that bigger geometric changes are more important than smaller ones. And that's not necessarily always going to be true. And so what we did is adapted this into a supervised autoencoder that we call DiffNets, right? And the idea is that, oftentimes when we're looking at variants, if we just do PCA or if we do an autoencoder, our data is, overlapping in the space and we can't see clear differences, right? So we switch this to a supervised autoencoder where we have the standard encoder and decoder, but in addition to that task, the neural network also has to predict a label from the latent space, right? So this could be something quantitative like activities for different enzyme variants, or just, zeros for the slow variant and 1 for the fast or whatever, right? And what this is going to do is force the latent space to dedicate more representational power to degrees of freedom that are important for determining that label, regardless of whether those degrees of freedom are, correspond to big conformational changes or slow or whatever other heuristic you might use otherwise. I was going to say what's important for that label, but of course you do need to get the geometry still for the autoencoder to work. One challenge, which is really interesting here, is that, of course, we don't know the labels for individual conformations. And so we also incorporate ideas from expectation maximization. So we start off with some initial labels where we say, okay, everything from this ensemble gets a 0, everything from this other ensemble, maybe another variant, gets a 1. But then we're going to update those to get a self-consistent set of labels. So structures that are similar will get the same label, but the variants will be biased somewhat towards different ends of the spectrum. There's a couple of other ingredients I won't have time to go into, but the great thing is that now we can go from having these overlapping ensembles to something where we still have overlap, because there are structures that are similar between the ensembles, but we hone in on these more subtle degrees of freedom or whatever that are important for distinguishing these variants, and can start to learn what's responsible for that. And so with very simple things, after training one of these autoencoders, like looking for interatomic distances that are correlated with the label that the network predicts, we can start to tease out what's responsible for these functional differences. And so in the case of NSP16, what we learned is that the more active variants have a larger distance between residues surrounding the active site. And then actually in the absence of NSP10, the active site tends to collapse and occlude substrate. And so what we imagine is NSP10 comes in and is allosterically stabilizing these more open structures that can accommodate substrate. And knowing that there's this allosteric here, now we went and applied a bunch of our pocket detection tools and said, okay, well, how could we take advantage of that? And what we found is that there's this really compelling cryptic pocket here in orange at this interface that can allosterically control how occluded the active site is. And importantly, it turns out to be present in all of the extant coronaviruses that we've looked at, and absent in human homologs. So this is great because not only can it impact function, but it can also potentially do so specifically for the viruses and not for host proteins that would give rise to nasty off-target effects. So we've got some new collaborations to go after this, and also looking at spike variants. And I'm happy to talk about other systems, but for now, I'll thank my group and everyone who participates in Folding at Home, and the many organizations that have supported us with compute power and funding.
Okay, thank you, Greg. So it was a very nice fascinating talk. So there are questions. The Pratyush, you want to ask yourself, or shall I read it out? Hello. Okay. So I will read it out. Pratyush is asking that is the fast dynamics reversible, at least in a state-to-state movement sense? That is, a conventional long simulation would eventually get unstuck, but also revisit previous states. Similarly, would fast come back to the previous state, or does it only run forward?
Yeah. So it will revisit states, right? Especially as given the roughness of the landscape, it's going to get stuck. And now the exploration term is going to cause you to go and revisit states where you have less data. It is one thing that we have found, especially for more dynamic systems, though, like partially disordered proteins, is that it helps to run fast multiple times rather than just running it once for a longer number of generations to help make sure that you do get heterogeneity in the pathways. But for things like beta-lactamase and lots of these viral enzymes that are pretty globular, even one pass has been quite good. And we've gone and run fast, with order of 10 microseconds of simulation, and then we've gone unfolding at home and gotten a millisecond of additional data. And the thermodynamics and kinetics don't change much, and we get really good agreement with experiments in the subset of systems where we're doing that too.
Okay. So Jagannath is asking that for spike protein simulation, he wants to confirm that whether the movie that you have shown are the actual movie or the trajectories that are stitched from MSM.
That's from our MSM actually.
Okay. Ravi wants to ask, Ravi Radhakrishnan wants to ask you that whether you have looked into the recent Omicron variant, given that you have looked at so many different.
Yes. Yeah, so we have been running simulations of Delta and some of the other variants, and we just in the last couple of weeks started up on Omicron. So we're excited to see, whether these patterns we saw with the different spike homologs, hold up, and if we can automate this to help, make some predictions about what other new emergent variants might mean for transmissibility, for example, since sadly Omicron probably won't be the last. Hopefully it's
yeah. So I have a similar question in the same line. That's why I'm asking. So you have shown 3 variants of COVID, and as you see that with more and more mutations, they're kind of getting lesser and lesser fluctuations. So the peak is coming towards the left side, so lesser distance separation. So does it mean that it is going to have a stronger binding, or does it lose some kind of stability or flexibility?
Yeah, so there's two major things that we have been paying attention to, right? So one is that the more closed things are, the lower your affinity for ACE2 is going to be, which is going to interfere with cell entry and transmission of the virus. But the other sort of orthogonal factor to consider is how strong the affinity between an exposed receptor binding domain is in ACE2, right? Because you can have the spike be more closed, but if when it's open, it binds more strongly to ACE2, your net affinity is going to be the same, for example. And so we think that these 2 factors can go a long way toward predicting how spike mutations are going to modulate cell entry.
Okay, great. So I think there are a couple of more questions about DiffNets. Maybe I will just read out. Atri is asking that is DiffNet similar to Gaussian mixture autoencoders, where the loss function depends on the number of clusters as well?
You know what? That's interesting. I'll have to think about it. I mean, I definitely think, in the sense of being autoencoders, there's something there. But this supervised component is going to be different, I think, because it's going to, without that, any autoencoder is going to, tend to dedicate more representational power to large geometric fluctuations, large and higher variance. Whereas with it, you can resolve, more subtle conformational changes, for example, that you might have glossed over otherwise.
Okay. I think there was a last question by Abhishek Das. Is saying that nice talk. What was the distribution of the lengths of individual trajectories that you harvested from folding at home light? Like this is reference to your spike protein work.
Oh, good question. So I would have to check for spike. It's quite variable though. So with FAST, what we found is that, simulations in the order of 40 or 50 nanoseconds, in each generation are good because you have enough time to get out of the starting state, but not so long that you're losing control over where you gather data. So that's pretty uniform. Once we go to folding at home, it's kind of an exponential distribution, the simulation lengths. So we end up with a handful of, much longer simulations and many shorter simulations. So I think most of our simulations are at least 100 nanoseconds, but we do have some that are, microseconds even, I think, for the spike or approaching microseconds. And of course, they're started off all over the landscape. So they have a bunch of mixing so that we get the mixing we need to get, the thermodynamics and kinetics more globally.
Okay, I think that's all for now. Maybe you can answer the questions if there are more in the chat box. So it was great talk, Greg. So next talk will be at 9:20. So we have 5 minutes of break, and then we'll resume from 9:20 as the schedule says.
Yeah. Thanks, Arnab.
Is it okay, Nishan?
Yes, perfect.
To let you know, I'm on.
Yes.
Yes, Niranjana. So we'll start at 9:20. 5 minutes later.
See you soon.
Maybe you can try your screen sharing, Niranjana.
Yeah, I could do that.
Yeah, I think Anji Babu has to take. He has taken it out.
Okay, let me try that. So, I'm clearly audible, I hope, right?
Yes. Yes, it is visible. You can make it full screen and then. Yeah.
Does it work?
Yeah, fine.
Okay, great.
It's perfect. Yeah, thanks. Yes, sir. Now we can start.
Okay.
Okay. Yes, I was mute. Sorry. So so welcome back to after break session. So the first talk will be given by Professor Niranjana Gupta from IISER Kolkata. So Niranjana, you have 30 minutes for talk and 5 minutes for questions.
Thank you very much, Arnab. I hope the screen is visible because I had to quit and enter. Okay, thank you very much. And thanks also to the organizers for, this is the first time I'm speaking in RARE. And it's actually, a great pleasure to be here. So thank you very much. What I want to lay out today is more of a journey than perhaps any end results. We are actually new to these, we are actually starting out. So I just wanted to relate to you, our journey over the last few years towards, building, some efforts towards building algorithms which will hopefully help in the enhanced sampling of biomolecular systems. So before I begin, I just want to say that the people who have accompanied us on this journey are quite a few. This is a figure taken, let me just go to my laser pointer. This is our group lunch a little bit before COVID started. So, there are some recent and past group members. It's an admixture. Some people have left, some are still around. But this journey started when Paul, who is right now a PhD student in my group, came to me. He's sitting right here. He came to me in, I think, end of 2016 and expressed that, he would like to do some kind of computations for biomolecules. So we started off, and coincidentally at that time, I had somebody visit me after a talk, who was actually working in space sciences. And, I was just told that, look, you have so much data, you should start thinking of using Bayesian statistics. So I kind of introduced Pallab to this person, and the journey kind of triggered extempore over there, without much of a planning. But I think we have come a bit of a way since then, and I'll try to lay out a journey. Generally, in my group, we are interested in understanding the behavior of biomolecules under a variety of conditions. Okay, so this is a schematic of an energy landscape. And essentially, we are interested in understanding that if we subject these states to a variety of perturbations, such as what you have in a real biological environment, what do you expect? So along the way, and of course, we are also interested in, estimating the transition rates between different states. So I'm highlighting some of the states that we are interested in. And along the way, we have looked at how these perturbations in the form of solvent crowding, in the form of surface interactions, and when you have extreme conditions, when you have structural modifications, either chemically inside a cell or imposed otherwise, how do these states respond? So in the past, we have looked at the, how surfaces affect the free energy landscapes, and we concur that the role of surfaces can be ambiguous. Very more recently, we have looked at how surfaces, which are confining surfaces, can have an effect on biomolecular conformations, and how do the stabilities change. We do find that if you do an ultra confinement, you can actually change the ratios of the native and the folded and the unfolded states. We are also interested in what happens when you subject protein assemblies to extreme conditions. So we have been looking at the so-called cold denaturation or cold dissociation, and can we generalize some of the results? So, and very more recently, we have started looking at how the solvent affects these interactions. So in different ways and forms, we are interested in biomolecular response anyways. This is also an example of a recent work where we are looking at how the non-structural protein of a protein which is essential for Zika replication. What happens if we force some allosteric conformational changes there, right? How do we expect the free energy pathways to change? So these are kind of a little bit of a wide overview of what we do, but essentially we are interested in determining the free energies of transformations under different conditions. As a function of what kind of reaction coordinate, that's an open question. That depends on the actual system that we are looking at. But it, it's, and this audience is not, I need not underscore this for the current audience, finding these can be extremely difficult and also computationally challenging. So, if you have limited resources, this journey itself is quite arduous. And the open-ended questions are, what is the location of the transition state? Can we find, suitable descriptors? What are the magnitude of these barriers? And if these three questions work out, then can we actually reproduce some transition rates very satisfactorily? So these are very open-ended questions. And what we found is, what we kind of started working on is, we started using some very simple ways, statistical ways, to at least find the heights of the barrier heights of the transition states. Okay, so as we have been hearing, since this is the second day of RARE, and this is what the RARE conference is about, I understand, is, can we discover novel methods? Can we go into efficient determination of, can we efficiently determine, can we kind of come up with learning-based algorithms? Can we find efficient ways of looking at these reaction coordinates? Essentially, can we complete the free energy picture here? Okay, so we have come up with a rather simple algorithm, which we name expectation maximized molecular dynamics, and I will discuss with you, what are the scenarios in which it works, and what could be the potential limitations, and to what extent can we push it? Can we really take it to more complex systems? So again, this is something that needs no introduction to this audience, is that when we have an underlying free energy landscape, and there are certain degrees of freedom which are easy to sample, we can actually sample these. If these barrier heights are, just a few kcal per mole, perhaps we can sample this without any biases. But then we do have some missing data. But somehow, if we can complete the entire distribution, we can actually attempt to, get a picture of the free energy differences. So what we resort to are Bayesian methods, okay, where we actually assign certain distributions, okay, to the unknown states. And if we can, in a probabilistic manner, if we can determine, which are the best distributions which can give us these, which are the best fits for these distributions, then we can perhaps complete this picture. It's kind of diagrammatically explained here, assuming that the distributions are Gaussian. Okay, so again, if we have a set of Gaussians, we can find the relative probability that a certain data point comes from a certain Gaussian. And if we iterate it, and if we update the Gaussians, we can determine, the best way in which this entire data can be modeled. And once we complete the entire distribution, we can actually get the free energy differences between the corresponding states. So what we also did was, we actually coupled the expectation maximization algorithm to what we call necessary and sufficient sampling algorithm. Okay, so even for the minima, even for the positions of the minima, we do not want to waste time. We do not want to waste computational time doing unnecessary sampling. So if we use this two ways, it overall increases the efficiency. So again, I'm just laying it out in a flowchart kind of way, but essentially, if we use expectation maximization both ways, we should be able to reduce the sampling time effectively. And that's something that I'm going to show. What we also discovered along the way is that if we have periodic collective variables, torsion angles, for example, Gaussian is perhaps not the best way to sample this, and a very related distribution known as the von Mises distribution, where you use the modified Bessel function of order 0. Okay, this k is kind of an inverse of the standard deviation. Mu is a representation of the mean. If we use a function of this kind, then we don't have to deal with complexities at the boundaries for periodic functions. So using this, with this information in the background, we started with very simple systems. I'm just looking at proline isomerization here. And as a benchmark, we used a very conventional method, adaptively biased molecular dynamics. And we start with 3 independent simulations with the EMMD method, with the necessary and sufficient sampling incorporated. And if we do that, we find that if you compare it with your benchmark ABMD, with the EMMD, with the one Mises distribution, we can actually fairly satisfactorily at least sample the cis to trans isomerization. So that worked very well. So this was the first point, and it was quite encouraging for us. Again, but we found that if we use the Gaussian mixture model, it doesn't work very well. So depending on what kind of collective variable you have, it's a better idea perhaps to switch to the other distribution. Now, if you again look at alanine dipeptide, again, perhaps the hydrogen atom of rare event sampling, we find again that, if you're looking at this and if you are able to actually assign the minima, okay, that you can do with standard sampling, we again used expectation maximization to kind of restrict the amount of sampling at the minima. What we do find is that, again, referencing with ABMD, we find that we can sample more or less the correct energy landscape with roughly 2 orders of magnitude less sampling time. So the efficiency is significantly increased, and one could argue that the free energy landscape is not extremely off. Okay, of course we are not perfect, but again, this is one method. Perhaps with other methods and with even higher sampling, you could putatively get to a better representation. Again, then we tried it with this particular protein, beta-2 microglobulin, and it has been experimentally shown that at position 32, where you have proline in the cis form, if you start fluorinating that proline, the cis to trans barrier is effectively changed, is altered. Okay. So we tried to reproduce this. We fluorinated, we did not do it for the whole protein, but we took the fluorinated form of proline, and we found that, if this this order is, can we reproduce it with the EMMD method? And we find that we do. However, when we start fluorinating, the absolute error between the reference method, ABMD, and EMMD, it starts increasing. Now that could be perhaps because, we could use better force fields, perhaps because, the fluorinated state with perhaps a different method other than ABMD could have done better. We don't know. But what we do find is that the order in which we get this is reproduced with EMMD. So that again is quite encouraging. And again, what is really interesting is if we use simple CPUs, for those of us who do not have access to extreme computational resources, we find that, EMMD is encouraging because, we can use at least 2 orders of magnitude less computational time and more or less converge into the free energy transition barriers that is relevant for a given system. Down the line, we have also worked with variational approaches where we do not use a fixed set of distributions. We kind of vary the distributions depending on, how the collective variable proceeds. But we also thought that we could use this the biases, we could more or less narrow down on the magnitude of the bias and perhaps use that information into a conventional sampling method and, come down to, not wasting any time into finding the magnitude of the bias itself. Of course, finding the correct collective variable for a more complicated system is a problem by itself, which I'm going to relate in our ongoing journey. So then we decided to see to what extent we could, push our limits. So it is okay to do it for proline and alanine dipeptide, but what if we try EMMD for an actual complex protein system? And that is something we started trying. So we have looked at one of these kinases whose conformation was reported very recently in its active as well as 2 inactive states. The ABL kinase belongs to the tyrosine kinase family. It has a lot of biological relevance here. And so you have cytoskeleton remodeling. It can respond to DNA damages, being a kinase actually. It belongs to the SRC family of kinases. You can have fusion proteins and it can trigger oncogenic pathways. So essentially we really need to know, what are the transition states between the active and the putative inactive states. So if we want to actually map down the key structural elements of this protein, it turns out that there's an A loop, okay, which I have highlighted here. There's a DFG motif. There's a phenylalanine right here. Okay, so you have a DFG motif, a short motif, which has a recurring importance here. There's a hydrophobic pocket here, and there's another hydrophobic pocket at this region. Okay, and it turns out, this is experimentally shown by Zhao et al., that the active state is the dominant state. Okay, it occupies about 88% of the ensemble, and then you have two inactive states. Okay, I'm going over this again. And essentially what happens between the active states and the one clear signature of the transition from the active to the inactive is the flip of this phenylalanine. Okay, we are showing here the flips. Now, this is kind of a summary of the key differences between the active and the inactive states. The A loop is open and all. The P loop goes from a kinked to a stretched form, and the DFG motif goes from the in to the out form. Okay, so we kind of started narrowing in on the DFG motif, and there are some key features of the structural transition. Okay, so what we started guessing around is perhaps the A loop backbone dihedrals, the A loop RMSD, the DFG movement, or the root mean square deviation between the relative RMSD between the active and the inactive state, or even the ion pair distance between glycine and lysine. These could possibly, or a combination of these could give us, useful collective variables, and perhaps we could go back to implementing EMMD after that. Now, it turns out that if we design a collective variable of this kind, where we simply look at the center of mass distance from the DFG motif to the hydrophobic pockets 1 and 2, we kind of reproduce, certain signatures of what is available experimentally. So CVDFG, we call this CVDFG. It's a scaled difference, between D1 and D2 here, okay, divided by D0. CVDFG clearly differentiates the active and the inactive states. And, what is not shown here are the corresponding values from the experimental data, from the NMR data, and it turns out that if you estimate the CVDFG, they sit right there. They sit right between the distributions that you get from direct sampling. So we thought that CVDFG would be a good idea to, a good starting point for the collective variable. Again, if you project the distance of CVDFG from the hydrophobic pocket or the leucine 373 from hydrophobic pocket 1 and leucine 267 from hydrophobic, the central motif of CVDFG from these two pockets, we find that, that clearly demarcates the active, inactive 1 and the inactive 2 states. Then, if you start doing a conventional sampling, starting either from one of the two inactive states, and you believe that you should be able to, go to the active state, using these as the collective variables, you find that, it actually depends on your starting state. Perhaps it's an artifact of insufficient sampling. These are results from ABMD simulations. Perhaps we need to go further in sampling. But clearly, depending on where you start from, you either end up in the active state or you do not. So clearly, even these collective variables are inadequate. And actually, if you look at the state that you end up with over here and look at its RMSD with respect to the experimentally reported active state, you find that you come up with huge RMSDs. In other words, even if you hit upon these collective variables, even if you hit your target, there are other degeneracies in the collective variable space and which you are unable to model with this simple collective variable. So there are significant deviations from the experimental states if we take this approach. So obviously we have to improve our approach. And what we did after that is that we started looking at the projections of CVDFG into the other collective variables, into the other, and possibly coming up with other reaction coordinates. So, if we use independent trajectories and if we use variational Bayesian Gaussian mixture, again, our journey was a little bit of a trial and error. So what we find that if you look at the projections of the DFG into different states, what you will end up getting is, if you really introspect the extent of correlation of CVDFG with some of the torsional angles involved with the other pockets, we are looking at alanine 399, and it turns out that it is extremely well correlated with CVDFG. And if we look at lysine 419, okay, it is actually much less correlated with CVDFG. So it's possible that these two, themselves could be a combination which would map out the conformational space of this protein. And it turns out that that guess may not be a very bad idea. So again, we have to switch back to von Mises because, Gaussian mixture models is perhaps not the exact way to do it. So a combination of these two CVs perhaps will be, one way forward. We could perhaps map out the inactive 1, inactive 2, and the active states. With this combination, we are still searching. Okay, so we also started looking at, using the simple root mean square with respect to the active state. Is that going to be a more appropriate way? Can we use a combination of this particular collective variable with some others? So that is also something that we are looking at. And we are also looking at the ion pair distance. If you have lysine 290 and glutamate 305, which is in the alpha helix, it's an interaction which is required for catalysis. And can we construct variational Bayesian GMMs over that? That is also something that we are looking at. And we don't know yet what is the best combination. But, hopefully, if we look deeply enough, we'll be able to come up with a combination which will actually give us a suitable mapping up of the entire degrees of freedom. That is also again something that we are looking at. But again, we do feel that given the importance of this particular collective variable, the CVDFG, okay, given that this particular flip is so much of a key change, a key signature of the transition from one state to the other. Something which is related to this conformational change is going to be a good signature of the transition from active to the inactive state. And we need to keep looking at the magnitude of change with respect to the experimental reported values. So with that, we are hopeful that we will be able to find the transition state barrier, even the conformations, not just the heights of the intermediary states that has been experimentally reported between the active and the inactive states of this particular kinase. And we are also working with certain collaborations here in IISER to, extend this to other kinases as well. Okay, so actually I'm done a little bit ahead, so that saves me some time. But in summary, EMMD is a barrier estimation methods for systems with known stationary states. It is actually manyfold more efficient compared to traditional methods. It at least gives us the height of the barriers, in a very quick way. And once we have the heights of those barriers, we can use that as a feedback into, informing the biases for more conventional methods. It could be potentially efficient for systems with more degrees of freedom, as we saw with the kinase. But then we have to actually find out, which collective variables are going to be exact. We have to actually find the degeneracy, and we have to find, which are the collective variables which could be discarded and which are the ones which should be retained. We can provide bias estimates for conventional enhanced sampling. That's something that I said. Potentially efficient. This got repeated. And again, if we could combine it with a smart CV search with some of the algorithms which are being presented in the RARE conference, that is something that we are looking forward to. If we could combine EMMD with other efficient CV search methods, then that could be, that could give us something which will take this entire method forward. And we are also looking at, extending EMMD to multiple dimensions. We could use variational approaches to automate model selections for more complex systems. So with that, I've come to the end of the talk. And of course, I first thank you for your attention. And I also thank the organizers for giving us a platform to present our very nascent work on this in this domain. We are still burgeoning in this direction, but we hope to take it forward as we go along. And once again, this work would not have come to this form had it not been for Pallab and Moitro and that initial discussion we had a few years back. And as we continue our journey, we look forward to more interactions with the platform here. Thank you very much, and Arnab.
Thank you. Very nice talk. So there are a couple of questions I can see in the chat box. So first question is by Anand. So he's asking that do the Gaussian or von Mises mixture distribution models take into consideration of the slowest CV, or the method is independent of that?
For the time being, the method is independent of that. We have to say that because it actually depends. You have to supply a CV at this stage. We are not, and since we supply a CV at this point, we are not at that late stage of a development where we can actually say that this CV is, represents the slowest degree of freedom. We are working on it, but for the time being, the way EMMD stands as of date, there are other developments going on, and there are some more students who have started working on this. But at this stage, this depends, on the degree of freedom that we select. So it does not necessarily represent the slowest degree of freedom.
Yeah. So in the same context, when you get this particular barrier height for a given CV, does it match with the conventional methods that already are known?
Yes. So at least for simple systems, as we saw for alanine dipeptide and proline, it does very well. But again, we are trying to be a little bit more ambitious here. We are trying to map it out with kinases. We are also actually, I did not cover that story because it's not even halfway. We are also trying to map it out with perhaps, proteins that are prone to self-assembly. So we'll see, perhaps we could hopefully be able to take it to a stage where, it will be, it will match the conventional methods better, and A, and better, and B, hopefully it will also be representative, or it will also capture the slowest degrees of freedom.
Okay. So next question is by Dhiman Roy. So he's asking that if you use the experimental related population of different states as the parameter in your model, can you calculate the population of the transition state and barrier heights?
So that is exactly what we are trying to do here. So, this is, we are working with, there's some a priori knowledge at this stage that we do need. We need to know we need to feed in the absolute free energy difference between the so-called minima. So what we want to find is the relative barrier height. If you go from state 1 to state 2, what is the height that you have to cover? So that is exactly what we are trying to find. Okay. We cannot at this time, find the minima themselves. So this is more about estimating the barrier height, which are separating different minima in the energy landscape.
So Jagannath has a comment saying that I agree with you that EMMD can be a great tool if CV search aspect can be explored with other similar techniques or smart techniques. That's what his comment is. Okay, so next question is by Aparna, saying what is the barrier height obtained for disulfide reduction, and which will be the best CV possible to address this reaction?
Aparna, we thank you for the question. That is something that we need to do. We haven't tried that yet. So in that particular work where EMMD was not used, I put that slide because I wanted to kind of convey our journey. With all the things that we do, we frequently encounter situations which need extensive free energy sampling. And most of us do not have access to extreme resources. And hence that was an inspiration to build a model or a neater method like this. So we really would like to understand that when I'm creating a perturbation and we are going from, a more integrated state to a more dissociated state of, say, the NS1 protein of Zika virus. Okay, so what is going to be the barrier height? That is, a few people have told me that that could be another system on which to try EMMD, but we haven't come around to that yet. And we really don't know. But I think I cannot make a wild guess, but it is going to be at least more than several KT. That is my guess. I really don't know the exact answer yet.
Okay, so thank you, Niranjana, for a very interesting talk. I think
Thank
if
you,
you have, if there are more questions, you can address them in the chat box.
Okay.
So we will now move to the next speaker. I know we are 5 minutes early, but that's okay. So, Gaurav, are you around?
Let me stop.
Yes, very much around.
Yes.
Great. So I'll share
yeah, you can share your slide. So next speaker is Gaurav Goel from IIT Delhi. So he's going to talk about linear response-driven molecular dynamics for protein structure prediction and protein conformational transitions. Yes, Gaurav. So start. Yeah, it is visible. And
perfect. So first of all, big thanks to the organizers to make it possible and for inviting me. And I know it's been a long time in making, but finally, I think even virtually we have been able to be together. Okay. All right. So today I'm going to talk about kind of very recent work from the group. So this has been, let's say, last 6 to 9 months kind of development. So some of the things we also don't understand, and hopefully we can use this opportunity to learn together. Okay. So broadly, what I'm going to tell you is that we have combined the linear response theory with what is known as excited normal modes molecular dynamics simulations to push conformational transitions on small timescales. And then we also show that we can use the knowledge of these intermediates which we are identifying on the transition pathway to build the free energy paths using, for example, path metadynamics. Okay, so that's the broad kind of theme. And in terms of two broad applications I'm going to discuss today, one would be protein conformational change on ligand binding, and another would be transient pore formation in a lipid bilayer. So I covered the outline. So that's what I mentioned. So I'll first start my talk with a little bit of method development details, and then talk about these two applications. So before we go to the problem at hand, the method development, although again, pretty much everyone in this room would be already agreeing with me that these are important questions which we are asking. So broadly, I will tell you what were our kind of motivations or interests in developing this method. So number one, I also want to know the intermediate. So if I'm looking at the apo to holo transition pathway, so if I go from the unbound to the bound state on ligand binding, in addition to the end state, I also need to know what are the intermediates. And I'm showing you one example here for the ribose binding protein, where actually it is one of the intermediates which is involved in ribose release. Okay, so these functional intermediates are present in several transition pathways, and therefore their knowledge is important. Okay. Second, on top of this, I want to get a complete understanding of equilibrium dynamics. So I need to know the structure space, the intermediates, how they look like, and I also want to calculate the underlying free energy landscape. So minimum free energy paths, how the transitions are happening, what are the barriers we are encountering, so on and so forth. Yeah. And in terms of challenges, I think this has been discussed a lot. This is a large landscape, large time scale problem. Depending on, for example, if there are allosteric effects moving away from the ligand interface, many other residues are involved. In that sense, it becomes a global protein size problem. So large landscape, and time scales could be anywhere from milliseconds, even higher, which are not directly accessible from molecular dynamic simulations. Second challenge is I don't know the relevant degrees of freedom. So I want to add some bias. I want to do metadynamics, umbrella sampling, other adaptive sampling techniques, but I need to know a few relevant degrees of freedom that are necessary to drive the change from one state to another. So those are some of the challenges one will encounter. Okay, so now I will introduce the methodology. So it's kind of little bit technical in terms of how we developed this approach. So it is based on the linear response theory, which was first, or at least in terms of being applied to proteins, One of the initial examples was by Iicii and co-workers in 2005. And they developed the theory with the aim of a weak perturbation, but getting a large scale change in structure. Okay. So they wanted a large scale change. And in that limit, if I want a large scale change, I have to assume the perturbation force, which is represented by F here, is a weak force. So in our case, for example, if I'm looking at the binding of a ligand to a protein, this external force is the force by the ligand on the protein. Okay. And what I'm interested in here is getting to this displacement vector. And the second input to this answer is coming from the covariance matrix of protein fluctuations calculated in the unbound state. Now, we show that, in fact, if you're only interested in the direction of initial response, that means if you go to the small displacement limit, then this force F can in fact be a strong perturbation as long as you limit the size of the displacement. Okay. So we kind of extend that analysis and show that if I'm only interested in direction of initial response, I still get the same expression, small change, but in terms of the unit vector, there is no change. So I get the same response direction in the limit of small displacement. That is if I'm only interested in the direction of initial response. So now we make use of this knowledge. So I have obtained this initial direction. Now the second part which we add is that we are going to find out the normal modes. And essentially we are going to map the response direction on the normal mode space. Okay. So we first do the parameterization of an isotropic network model. And for simplicity, we take all spring constants as equal. So this is kind of a well-established method. It has been used a lot. And typically one uses the fluctuations of protein atoms to get the parameters of ANM. So on the top left picture, that's what I'm showing, that we are using the fluctuations of protein atoms, and the anisotropic network model we are developing is able to reproduce those fluctuations. So that is very nice. Second addition which we made in the parameterization stage is that I first identify, so on the right, I'm showing you this one picture where I'm showing you what are the dominant modes along the response direction. So this is showing the weights. So I'm showing you the weights of the dominant modes along the response direction. So our second aim in the parameterization was that I want to reduce the number of these dominant modes. So we use a Shannon entropy measure, which would be equal to 1. If all the modes are contributing equally, it will be equal to 0. If only one mode is contributing to the direction, that means a single mode is completely overlapping with the response direction, then you get a Shannon entropy measure of 0. So in that sense, I want as small a value of the Shannon entropy as possible while preserving the reproduction of protein fluctuations. Okay, so we use these two constraints, and the benefit of this is that I get a small number of relevant modes rather than a very large number of relevant modes. So on the right, I'm showing you for one particular case of adenylate kinase, and depending on the ligand, in this case, the ligand is adenosine diphosphate. So when this ligand is binding to the protein of interest, adenylate kinase, we are identifying the 3 dominant modes, which I'm showing by blue arrows. And these are the modes which we identified in the initial structure. And it is, so broadly, this protein has 2 domains, the lid domain and the NMP domain. And the initial motion in other works also has been predicted to be in the lid domain. And that's what we are also predicting here, that the initial motion is in the lid domain. Okay, so I will come to this point again, that why I'm calling this initial motion and what else we do to get the final structure. Okay, but for now, all of these calculations I'm showing you in the initial unbound state, and therefore all these directions which I am identifying are only relevant for the initial motion of the protein, not the overall motion of the protein during conformational change. Okay, now the last task is I want to enhance the sampling along the response direction. And that is where this molecular dynamics with excited normal modes developed by Costa and co-workers comes in. So they have shown that you can select normal modes, and along whatever normal mode you want to bias the sampling, you can add an extra velocity term. Okay, so here that is what is this showing. I add an extra velocity term along the choice of my normal modes, and in this, the two parameters which will govern the direction and extent of conformational change are listed here. So one of them is alpha I, which we use to control the direction of conformational change. And as mentioned here, the direction should be along the response direction predicted by linear response theory. So alpha i's we will choose in such a fashion that the direction of conformational change in this excited normal mode simulations overlaps with the predicted direction by linear response theory. And the second very important factor is the extent of conformational change. So several other studies have already shown that the linear response theory is not accurate for large displacement. And even we are showing in our derivation that only in the case of small displacements, I can get the linear response theory expression, even in the case of strong perturbation. So we want to limit our delta t so that I stay in that so-called small displacement regime that is within the region of validity of the linear response theory. So those are the two, let's say, governing principles we are going to use to find out alpha I and delta t. So alpha I, as I mentioned, I want to ensure that the addition of kinetic energy is happening along the direction predicted by linear response theory. So for that, we use this simple expression which shows the conversion of kinetic energy added to a mode into its displacement. So we use this expression in very simple terms. It's clear that if I add the same energy, the displacement would be different depending on the stiffness of the mode. And the stiffness is being captured by the frequency for that particular mode, omega I. Okay. So if the stiffness is smaller, you will get larger displacement. If stiffness is larger, you will get smaller displacement. So intuitively, it makes sense. And mathematically, this is the equation we use for conversion of kinetic energy to potential energy to find out alpha i's. So with this choice of alpha I, omega I times OI. OI was my overlap factor. Okay. So OI gives me the overlap of response direction with any given normal mode. So it is a projection of response direction on any given normal mode. And omega I is the frequency for that normal mode. And what we are showing is that if I choose my alpha I not equal to OI, that is the overlap, but rather the product of overlap with the frequency, the change in the displacement is overlapping very nicely with the direction of linear response. So this we are showing in our paper, which is under review. I'm not showing those graphs, but it was clearly shown that yes, with this choice, you are getting very nice overlap with the direction of with the response direction, which is the direction predicted by linear response theory. Now the second parameter. So I want to ensure this delta t gives me small displacements. So I will just read you or walk you through this plot here. So what I'm showing you is the structural change along the response direction, which we are measuring in terms of this parameter dp. So dp is essentially the actual change and its projection along the response direction. So whatever is the actual change in the structure, we take the projection along the response direction, and that gives me the dp vector. So this dp I am estimating at various different values of delta t. So larger the value of delta t, larger the energy, kinetic energy I'm adding to my response direction, larger will be the structural change. However, it might or might not lead me to the correct end state. So the green, the black plot here is showing you that as you increase delta t, dp in general will increase. However, I am also showing you in blue the Rmsd of any particular structure with respect to the end structure. So in this case, I'm interested in finding the hollow state structure. So this blue line is showing you the Rmsd of any structure with respect to the hollow state structure. Okay. So what we observed was that up to a certain delta t, these two variables, that is dp and Rmsd, we also looked at some other variables, for example, global distance test metric. So all these structural change metrics had a very nice correlations with dp up to a certain value of delta t, and beyond that point the correlation decreased, which is again coming back to the failure of linear response theory at large displacement. So all we are doing here essentially is an automated selection of delta t up to the region of validity of linear response theory. So in earlier applications of linear response theory, it was selected to a finite value, 2 angstrom, 3 angstrom, whatever worked. Here we are showing that you can actually obtain this on the fly by using this dp metric, and the region I have highlighted marks the plateau region, the first plateau region in dp as a function of delta t. Okay. So this highlighted region, so it's clear that if delta t is very small, the structure will go back to the initial state. So dp will be 0. So I don't want that. So whenever dp changes by a finite amount, the first finite change in dp we choose as our delta t. Okay. So in this sense, we are limiting the structural change in our any one iteration of excited normal mode simulation by design. We are making the structural change small, which clearly means that if I want to obtain a large scale structural change, I need multiple iterations. So those are the two essentially parts of this process. In each step, I make a choice of alpha I and delta t to ensure I stay within the region of validity of linear response theory, and I make a structural change which has a high overlap with the response direction. And secondly, now I will affect multiple iterations, and one main benefit of this multi-step process is that in addition to the end state structure, you also get information on intermediates. So the target was get the information on intermediates. This multi-step protocol automatically allows me some intermediates. Are those intermediates good or bad? We'll discuss later. Finally, I need to stop somewhere. So we use the protein ligand binding energy estimate as a marker of our stop point. So this is a combination of two contributions. One is the MMBBSA binding energy. So this is an implicit solvent method to calculate binding energies. And second is that we also add the protein strain energy. So as the protein structure changes with respect to the unbound state, we add a measure of strain energy, which gives me the total protein ligand binding energy in this case. Okay. And what we see here is, in this particular case, for adenylate kinase adenosine diphosphate, after 6 steps, I get a minima in this metric delta delta G, and therefore we stopped at this point and we said this is my prediction for the holo state. So this we tested for 3 systems which presented differences in the extent of structural change, the amount of non-linearity, which is mainly coming from presence of multiple modes or presence of structural change along multiple modes, and finally also presence of allosteric effects. For example, the ribose binding protein shows prominent allosteric effects during its structural transition. So in each case, we are seeing that we are starting at a high RMSD with respect to the holo state structure, 7.2 angstrom for adenylate kinase, 6.4 for ribose binding protein, so on and so forth. For the last one, it was small. And in each case, we are able to come very close to the holo state structure. So almost within 2 angstrom of the holo state structure. We also found out the modes along which the motion is happening, comparing the crystallographic structures and our predicted structures. In both cases, we see a very nice overlap in respect to displacement along each individual mode also. So now the next part, which I mentioned, that in addition to the end state, I'm also interested in finding out the information on intermediates. So this I will cover in less detail. The student who worked on this part, Rajat Punya, he is also presenting a poster. So he will cover these aspects in more details in his poster. So I'm just going to cover the key points here. So one, we compared the path we are finding in terms of just structure. Right now, I don't have an estimate of free energy, but I'm saying that whatever set of intermediates I have identified, my protocol is leading me to a minimum free energy path. We will test that later on. But right now, the assumption is that whatever set of intermediates we have identified, they actually lie on the minimum free energy path. And this we compare against, for the aryl kinase problem, we compare against bias exchange metadynamics. This was done in the set of 2 coordinates, this bias exchange simulations. This we have taken from Lee et al. Paper, Biophysical Journal. So that is shown in red. And the prediction from our multi-step protocol is shown in black. So what we are seeing here is that overall matches. Maybe the bias exchange simulation missed an important coordinate. Therefore, the transition is not so smooth. When we add one more collective variable, I'm not showing data here, the transition looks much smoother in our multi-step MDENM protocol. So if I add the third mode also in the space of 3 normal modes, the transition looks much smoother than what is being shown here. This is a 2D projection of that 3-dimensional transition. And roughly it matches what was predicted. Okay, I will not go in much details. Now on the right hand side, I am showing you the comparison of intermediates we had identified for the ribose binding protein. So we compare our intermediates against crystallographic structures in terms of RMSD. And I'm also showing on the right the path tracked by these crystallographic structures, as well as the path tracked by umbrella sampling simulations taken from Ravindranathan and co-workers. Okay, so again, what we are seeing is the set of intermediates we are predicting are matching very well with other more detailed, more expensive simulation techniques, as well as whatever crystallographic structure data was available. Okay, now finally, what we did was we took these structures as inputs to a path metadynamics simulation to find out whether the path which we are predicting is indeed correct or not. That is, the set of structures we have predicted, do they lie close to z equal to 0 or not? Okay, so here I'm only showing you the answers for the hollow state. We required around 500 nanoseconds of simulations to get convergence. For both apo and hollow state, the answer was almost similar. So we used the same set of structures for the apo state also, but of course we did not include the ligand in that simulation. And this hollow state simulation, we included the ligand. So what we observed in fact was that if I do not include the ligand, then the state near s equal to 0, which is the unbound state, was the most stable state. And when I include the ligand, you can see on the bottom left that the state corresponding to s equal to 1, which is the bound state crystallographic structure, is the most stable state. And the path which we are predicting is lying very close to z equal to 0. Also, what we can see from this picture that we are predicting 2 possible paths. And in fact, the bias exchange metadynamic simulations, the data I'm showing on the right, again from the group of, or from this earlier publication, that was also predicting 2 possible paths. And right now we are going to further, we are evaluating this second graph ourselves in the space of these 2 angles, the lid core angle and the NMP core angle, to find out whether the path predicted in that is the same as the path predicted by us using the path metadynamic simulations, where the structures were taken from the multi-step MDENM simulation. So that is kind of the first story for this talk today. The second application, as I mentioned, we are also interested in lipid bilayer conformational dynamics. And right now we have investigated passive pore formation, but going forward, we are also going to work on active pore formation, which is basically allowing us to determine the permeation of ligands, peptides, molecules across a lipid bilayer. So some of that we saw actually in yesterday's talk by Ron Elber, where they were looking at permeation of peptides, a specific peptide they had chosen, across a lipid bilayer. So in general, the collective motions in lipid bilayers are very diverse. Membrane fusion, pore formation, interaction with membrane proteins, cell penetrating peptides, so on and so forth. All of those will lead to a coupling of macromolecular motion with the lipid bilayer motion. Okay, so these are some of the pictures which are showing the various kind of motions. On the bottom left, you are seeing peptides transporting across a lipid bilayer, and on the bottom right, I'm showing you pictures of spontaneous pore formation observed in a lipid bilayer. This was done using, again, free energy calculations along a pre-decided set of reaction coordinates. But they show that even in absence of any ligand, any peptide, you can get pore formation in a lipid bilayer. So now again, what we do is we first try to develop an elastic network model for the lipid bilayer now. So protocol is almost the same. Only thing which we add or which we make different from the earlier work, which I showed for the protein ligand system, is that now I consider the environment also as part of my overall model development. So that means in addition to system spring constant, I will have two more spring constants, the environment and the system environment. And in this case, it was essential to do that because the overall elasticity of the lipid bilayer is much smaller than the protein molecule. So therefore, the interaction with the environment was considered to be very important here. And we actually find out that if I don't consider that interaction, I am not able to reproduce the fluctuations of the lipid bilayer. So if I ignore the interactions with the environment, I will not be able to reproduce accurately the fluctuations of the lipid bilayer. So the parameters we have is the cutoff distance RC for the normal mode development, the spring constants, the 3 spring constants. So total 4 parameters I have, 4 parameterization. So we used a 500 nanosecond molecular dynamics simulation of a 256 molecule lipid bilayer. Roughly the size of the bilayer in the XY dimension was 8.8 into 8.8 nanometers for the calculation of properties, which in this case is mainly position fluctuations, which we use as input to train our model. So again, what I'm showing on the left is that the prediction of fluctuations by our model in blue. I'm showing you the answer from 2 possible values of the cutoff RC, 10 and 14 angstrom. They match overall well with the predictions or the values obtained from molecular dynamics simulations. Okay, so this was again average over all 256 lipid molecules. Okay. Then the second variable in the equation, as I mentioned, is gamma SE also. So you can clearly see in this case, sorry, the variable is not very visible on the x-axis. So it is a ratio of gamma SE to gamma SS. So the spring constant for system environment interaction divided by the spring constant for the system interaction. So I can clearly see that if I take this number to be small, that if I take it to be 0, I get small values of position correlation. Okay, so here I'm comparing the Pearson correlation for position fluctuations between the model and the MD simulation. So if I don't include environment, I get a very small value of rho. But if I include the environment, and we are seeing that the maxima is coming approximately around gamma SC by gamma SS equal to 1. So if I include the environment, I get a very high correlation between the MD fluctuations and the prediction from the model. Okay, so therefore I include the environment in our answer. For RC, which is the cutoff distance which we are taking for bead interactions, we see that it is a very broad range. We take a value of 10 angstrom, but 10, 12, 14 work almost similarly. Okay. So on the left, I'm showing you the last piece of the puzzle, the model, where what is the effect of environment interactions. So I can clearly see here that because my solvent is essentially a Newtonian liquid, I will need to take a very small value of gamma EE. I am seeing it is approximately 10 to minus 3, 10 to minus 4, where you start to see a plateau. Okay, so I need to take a very small value of this variable to be able to reproduce the position fluctuations of the lipid. So main parameter to add was gamma ES, the environment interaction with the system. Okay, finally, to get the absolute value of the spring constant, we use the height fluctuation data on the lipid bilayer. Again, this height fluctuation data was calculated from the same MD simulation. And what we are seeing here is that for all wave vectors, a single value of this gamma SS, in this case, it came out to be 1 kilojoules per mole per nanometer square, gave an excellent match with the MD simulation data. So therefore, this kind of summarizes the set of parameters we obtained for our model. How many minutes I have?
Yeah, so you're almost like you have 1 more minute actually. For
One more minute. Perfect. Thank you. So I will skip over this part. So this is just kind of validation.
But if you can take some 2-3 minutes more, because I'm 35 minutes will take 5 more minutes to finish. But if you want discussions, then you have to cut short.
Yes. No, I will wrap up. Thank you. I will take one or two more minutes. Okay. So I'm kind of skipping this validation part of the model. So broadly, we did the same things. We looked at the kind of motions we are getting from our model and the kind of motions we are getting from either experimental data or from molecular dynamic simulations. The one addition with respect to the protein problem was that I also need to account for friction along the modes. So we also accounted for friction coefficient along the modes and solved the Langevin equations, which include both the elastic network model and the friction effects in the space of normal modes. And some of the properties like bilayer bending modulus, lipid tilt modulus, relaxation of the undulation. So this relaxation of undulations is where I need to account for friction effects. Otherwise, I will not predict them correctly. So all of these, broadly, we were able to match very well with the available data, either from experiments or from molecular dynamic simulations. Okay. And this one application, I'm just going to mention that it is possible. So using the same linear response driven molecular dynamic simulations, the main change was here I don't have a perturbative force. So to find out the response direction, rather than using a perturbative force, we define this order parameter, which tells me the shape of the bilayer. And then I use this argument that I choose a direction where the order parameter gradient along the modes is largest. So using that direction as my constraint, I find the set of modes which is relevant. The bottom right picture is showing you the velocity field in the initial lipid bilayer, which is leading to pore formation. And again, we do a multi-step simulation. So this is the top view of the lipid bilayer. Bottom molecules are not shown. So I'm able to see the pore formation, transient pore formation. This is the side view, which is showing you that the water molecules are able to penetrate in the lipid bilayer. And again, now the next task for us is to use these as inputs to a path metadynamics or adaptive sampling technique to find out the free energy profile for this transient pore formation. That's all. So all of this computation work was done by Rajat Punya, a PhD student in the group. He's also presenting a poster mainly on the protein structural change on ligand binding work. And for funding, we thank the SCRB board of Department of Science and Technology and IIT Delhi HPC Center. Thank you for your attention.
Yeah, thank you, Gaurav. So very interesting talk. So actually, there's a question by Shravani Tarottar. She's asking that, Gaurav, how does the linear response driven MD relate to path reweighting?
So I don't have an answer to that question.
Hello?
Yeah, I don't have an answer to that question. So I'm not aware fully about the path reweighting technique.
Okay. So Gaurav, so I have a couple of questions. So basically, in your elastic network model, how is it different from, let's say, standard like large elastic band approaches where we create a lot of replicas and then use a force constant between them? Is it similar?
So here I am. So if I understand you correctly, you are creating the bands between protein replicas.
Right.
But here this anisotropic network model is essentially a simple, let's say, approximation to the Hamiltonian of the protein. So this is a coarse grain. You can think of this as a coarse grain interaction potential.
Okay. And
for a single protein.
Okay. And when you are talking about lipid bilayer, you said that you had to also couple in the environment.
Mm-hmm.
So, but how large the environment size would be in order to get the right fluctuation? So how many? What will be the length scale around it?
Yes. Correct. So we use a single parameter RC for both the lipid bilayer as well as the lipid bilayer and environment interactions, which in our case we chose to be 10 angstroms. So 1 nanometer. So that was the let's say length scale of interaction of the lipid bilayer with the environment, which was sufficient to reproduce the fluctuations of the lipid bilayer.
Okay. And in the beginning part, like I saw that when you are trying to compare with metadynamics and I think bias section metadynamic simulations and your system, you were saying that they were missing some of the collective variables for the path. Can you just a little bit elaborate on that?
Yes, I mean, of course, we have not fully calculated the free energy profile, so it will be something which I'm not sure of. But what we are seeing in the in our space, I will go back to that slide. So there are, if you look at, for example, I1 to E1 transition,
Yes.
okay, and then E1 to E2.
Right.
So that entire thing seems highly non-linear.
Okay.
So this might be a real effect, or as I mentioned, that if I go to the 3-dimensional profile, then this looks much smoother. So here what you are seeing in the black curve, which is predicted by us, there are several bounce backs. The collective variable is increasing then decreasing again.
These I1, E2, they are all stable states?
They are all metastable states. And this I am showing you on the right. For example, we compared the intermediates which from our algorithm for ribose binding protein, and they showed very good correlation with the variety of crystallographic structures for this protein. Okay. So each of the intermediates corresponding to one particular crystallographic structure, and therefore we are confident that these individual structures are metastable.
Okay, got it. Okay, so I think I don't see any more questions, Gaurav. So thank you again for a very interesting talk.
Thanks, Arnab. Thank you.
Yeah.
So we'll stop screen sharing.
So we'll move it to our next speaker, Anupam Kundu from ICTS Bangalore. He's going to talk about large deviations in a class of long-range interacting models. Anupam, you can share your slides.
Okay. Can you see slides?
Yes. Slide is visible.
Okay, so let me start.
Let's start. Yeah.
Should I make it full screen or is it good enough?
I don't know. I'm fine, but I don't know about others. You can make it whatever is comfortable. Whatever is comfortable for you.
Yeah, full screen is good. Yeah, full screen is good.
Okay, so first I would like to thank the organizers for inviting me to present my work. Yeah, there's some problem with the full screen. Let me go to the this mode. Yeah, so there are some people. I'm getting some messages that some people are waiting in the room. Should I admit them?
No, you don't have to worry about that. We will take care of that.
Okay. So yeah, I would like to thank the organizers for inviting me to present my work, and I'll talk about large deviations in a class of long-range interactive models. And I think this talk will be slightly different from other talks, although within the theme of the broad theme of the conference. Okay, so I would like to acknowledge financial support from CEFIPRA and ACRB government of India, and also the computational support from ICTS. Okay, so let me start with rare events. So these are random events that do not occur often, regularly or typically. There are many examples. A simple example would be getting 48 heads in tossing a fair coin 50 times. Then there are other examples like high or very high or very low, extremely high or extremely low temperature in a day in a city, heavy rain, heavy traffic jam, large fluctuations like tsunami. There are many more. So these events are rare, but has important, often devastating consequences. So it's important to study them. So in probabilistic language, random events are described by probability distributions. And this is a typical schematic plot of a probability distribution. You see that there is typically there will be a peak and then there are tails. and around the peak there is a mean value, and around this mean value you consider a region of the order of standard deviation, and you see that the events within few orders of standard deviation have kind of appreciable probabilities, and we consider them as typical events because they have appreciable probabilities, and if you repeat the experiments some sensible number of times, you will find these events to occur. But there are events that occur at the very tail of this distribution, and they are rare. They are atypical events, and they occur with very, very low probabilities. And often these events, which are known as large deviations, these are large deviations from the typical behavior, like the mean around or the behaviors within standard deviation about the mean. So these events are very, very largely deviated from this behavior, and these are often described by probabilities of exponential form, not always, but often. And these exponential forms are more accurately described in terms of the cumulative probability, where this is a random event, a random variable, and we ask what is the probability that if this event, this variable has value greater than x, and that probability is exponentially small if that value is much, much smaller than the mean, and it approaches to 1 exponentially if that value is much, much larger than the mean. And this side is actually left. This L represents the left of the mean, and this R represents the right of the mean. And these are constants, these are called speeds, and these quantities, functions, are called large deviation functions. And these functions also appears in many, many contexts. One of the simplest contexts is sums of random variables. And this also appears in the simplest example is random walk, where you want to ask what is the probability of finding the worker which is doing a random walk at position M after step N. And if you compute this probability, this is a very simple problem, and all of us have done it. So if you compute it and if you plot it, and this will look like the distribution will look like this at different N. As N increases, the distribution becomes flatter. So this is at time at N equal to 20, this is at N equal to 30, and this is at N equal to 40. However, if we plot this distribution with some scaling, for example, if we multiply square root n on the y-axis and divide it by square root n on the x-axis, all these curves fall on top of each other. There is a data collapse, and the square root n is actually the standard deviation of the displacement of the worker. And this collapsed curve is actually Gaussian. So this property is mentioned by the fact that this distribution possesses a scaling form, and the scaling distribution is Gaussian. Good, very good. But this scaling distribution, this Gaussian distribution, cannot describe all the events. It can only describe typical events. For example, let us again consider a symmetric one random walk of step 40. A typical event would be to find the worker at position 6 after you make 40 steps, after the worker has made 40 steps. So that means it has actually taken 23 right steps and 17 left steps, and the probability of that event is 0.08, and that it can be predicted from this scaling function and pretty well. But an event, consider this event that in which the worker is at position 38 after the 40 steps. So that means it has taken 39 right steps and only 1 left step. And the probability of this event, if you do an actual calculation, is actually 3.64 into 10 power minus 11. Okay, but this scaling distribution gives only this. So this scaling distribution at very, very rare events does not give correct results, and they are only the large deviation functions, large deviation estimates of the probabilities give this kind of probability. So it's important to study these large deviations. So this, as I said, I was saying, these large deviation functions are form of the probabilities appear in many contexts. For example, in extremes of many random variables, current fluctuations across a junction, Cramer's problem of escape, queuing theory, Einstein's fluctuation theory, mathematical formulation of equilibrium statistical mechanics, finance, epidemic ecology, and many more. So let me in particular say that in Einstein's fluctuation theory, one considers a microcanonical system of energy E, and then if you consider what is the probability of observing a fluctuation in some observable, say, magnetization from M0 to M, this is actually given by this, and it is actually in the large deviation form, where SEM is the entropy per particle. Similarly, in canonical ensemble, the probability of finding a system in that energy E also has this kind of large division form, where the large division function is actually the free energy. Okay, so in this slide, let me kind of emphasize that the study of extreme events is actually different from the study of rare events. Study of extreme events actually asks questions about how big or small, whereas these questions, this asks actually questions how rare. So let me explain this in the context of independent and identically distributed random variables. Let us consider in such random variables, this question deals with what is the distribution of the maximum of this n variables. Whereas this question deals with what is the how anomalously large or small this x max, the maximum of these variables, how anomalously large or anomalously small compared to the mean values. So these questions are studied well in the context of independent variables. What about the correlated random variables? So one of the well-studied situation or very frequently studied situation in the context of correlated random variable, in which these questions are studied is random matrix theory, in which one considers n cross n random Gaussian random matrix. So you have a matrix and these elements are random, and these elements are chosen from Gaussian, and the joint distribution of these elements is basically this, which can be written in this form, where in the exponential you have trace of a dagger A is the matrix, okay, and in such a way that this distribution is actually invariant under transformations on the matrix, and these transformations actually gives rise to different ensembles. If this is invariant under Gaussian orthogonal ensemble, it's called Gaussian orthogonal ensemble. If this is invariant under inter ensemble, transformation, then this is called Gaussian inter ensemble, and similarly, if it is invariant under symplectic transformation, then it's called Gaussian symplectic ensemble. So you can choose elements like matrices from these ensembles and ask what is the joint distribution of eigenvalues or the spectral statistics. So this question was answered by Wigner in 1951, and he got this elegant result for this joint distribution of the eigenvalues. And this is the explicit expression where Zn is the normalization, which we know as partition function. And this is characterized by an exponent beta, by a parameter beta, which takes values 1, 2, and 3 corresponding to this ensemble GOE, GU, and GAC. But if you look at this expression closely, then you see that there is an interpretation which is given by Dyson in 1962. He said that look, this distribution can be thought of as Boltzmann weight of a gas of n pairwise repelling charges confined in an external harmonic potential in 1D, like this. And the repulsive interaction is this logarithmic interaction. So you can interpret that as a collection of charges sitting in 1D and confined in harmonic potential with this logarithmic interaction, like this. And then what is the density? And you can ask what is the average density of these charges. And Wigner showed that in the large n limit, this average density actually has a scaling form with this scaling function given by this, which is actually a semicircle, and this is known as the Wigner semicircle. And this suggests that these particles in the larger limit actually settle down over a finite region. It doesn't actually extend to infinity, it's over a finite region of space in the harmonic potential well, and that region actually scales as square root n. Very good. But remember, we are interested in the largest of these eigenvalues, which in the log gas picture, the particle picture, is the position of the rightmost particle. Of course, on an average, this particle will sit at the edge of the density, and it will fluctuate around this edge. And it turns out that this fluctuation is very small, which is n to the minus 2/3. And these are the typical fluctuation. And you can ask what is the distribution of this typical fluctuation. And this distribution was computed by these two mathematicians, Harold Widom and Craig Tracy. And they showed that this distribution, this typical fluctuation actually possesses a scaling form, or you have a scaling distribution, and the scaling distribution can be expressed in terms of the Hastings-Macleod solution of Poisson-Levet equation. Okay, very good. So this distribution, this scaling distribution, these are the details of this distribution. I'm not going into the details of this. This distribution describes these typical fluctuations of order n to the minus 2/3. But what about the fluctuations which are larger than this? That means order 1 distance away from the mean, like this side or that side. And these are described by large division functions. And And for this case, it has been shown that in the left side, the probability has this kind of large division form, the speed is n square. On the right side, it is e to the minus n times the large division function. And these large division functions phi plus and phi minus are explicitly known, explicitly the explicit expressions are known. I am not presenting them. Only interesting feature is that these functions, as you approach from this side to the mean value, or this side from to the this position, mean position. So this side means W approaches to square root 2 from left side is with square root 2 minus, and W approaches to the square root 2 from right side is W into square root 2 plus. And they behave as kind of power laws with some exponents 3 and 3 by 2. Very good. Now, if you look at this probability and consider taking this limit, this is the cumulative probability, and taking this limit and divide take log of that quantity and divide it by n square, and take a minus 10 and take n going to infinite limit, you can see that from this expression, if you take log of this, you'll get minus beta n square phi minus W, and this side you'll have minus beta n phi plus. Then you divide by minus n square, then and take n going to infinity, this side will go to 0. This will never anyway go to 0. Only this phi minus W survive, and you can find a function which you call which is a kind of analogous to free energy, and that free energy is like this, which is for W less than root 2, it has a cubic behavior, whereas for W greater than root 2, it is 0. And you can easily see that this function, if you take a derivative, if you take 3 derivatives, then at the third derivative is discontinuous. So by Ehrenfest classification, you can think of this as a third order phase transition. And this is not just a kind of for academic purpose, you consider this as a third order phase transition. This third order phase transition has also been observed in many other problems. For example, in chaotic cavity, you can see these third order phase transitions. Oilless communications, spin glass, where you try to find out the number of zeros in a random potential, not number of zeros, number of minimas in a random potential, and in that random potential you have a in the number you have a transition. Then entanglement in bipartite quantum systems, also you can see such transitions. Actually, these kind of transitions are first observed in string theory, in the large and gauge theory, and known as Gauss-Witten-Wardier transition. But okay, so this transition is observed in many systems contexts. Similarly, the Tessier distribution also appears in many other seemingly unrelated problems. Interface site fluctuations, longest increasing subsequence fluctuations in vicinal surfaces, etc. So this means that these two properties are kind of universal. So what we wanted to ask, how universal is these results if the interaction is changed? So remember that we had considered a gas of particles in which these are interacting by this logarithmic interaction. And suppose we change this interaction, then what happens to these results? So we consider this type of interaction in which the interaction is a power law, characterized by this exponent k and strength j. And we consider k to be greater than -2 for some reason, for some stability reason. I'm not going into the details of that. And you can see that this model actually captures a whole set of systems that have been studied earlier. In fact, it actually k equal to 0 limit actually gives rise to this model, which is log gas. For k equal to -1, one obtains one-dimensional one-component plasma model, which is a famous model that was studied by Baxter, and the many thermodynamic quantities of this model was studied by Baxter in 1963. Then k equal to 2 is the interacting integrable model, which is a famous model. And this also appears in many physical systems also, like in what is it called? I forgot that particular system. It appears in actually vicinal surfaces, fluctuations on the vicinal terraces, and other systems. This is not coming to my mind right now. For other values of k also appears in different situations. For example, dipolar gas confined in a 1D, and this kind of systems are also realized in experiments, like BC of chromium atoms or ultra-cold gas of potassium rubidium molecules. Then k equal to 4, 5 are also possible. Harmonic potential, we have considered harmonic potential. It is actually because it is most ubiquitous in cold atomic experiments. And also the absorption imaging techniques have been so developed that we can actually observe positions of the atoms or the ions. And such kind of Hamiltonians or energy function has been used in collective dynamics of many experiments. This is one experiment in which such interaction has been used, and the collective dynamics, collective behavior has been considered. And here the K effective was minus 2/3. This is another experiment in which they have created a chain of ytterbium ions, and they are able to actually create long range interaction with K of the form, this form with K between 0.8 to 1. And in this case, they can actually observe the positions of the atoms. So it is possible to actually create the density profiles or look at the age fluctuations, the kind of questions that I was talking about and I'll be talking about in more detail. Okay, so we consider the system is that we consider harmonically confined rich gas described by this energy function in thermal equilibrium. So the which is described by this Boltzmann distribution. And let me ask the first question is, what is the density for general K? And it turns out that for general K also the particles actually settle down over a length scale Ln, which is independent and also possesses a scaling form. And the scaling function is given by this. So y is x by ln in this. So the scaling function is given by this, which is characterized by an exponent gamma k. Gamma is an exponent which is dependent on k. And Ln scales with n as n to the power alpha k, where alpha k is another exponent which also depends on k. And this is a length scale of order 1, which also depends on k, obviously. And this gamma k actually characterized how the behavior of the density profile at the edges. And so these two exponents actually, alpha k and gamma k actually characterize the density profile. And you can get a whole different set of density profiles for different values of k equal to -2 to k going to infinity. And you get different shapes of the density profiles. And you can get shape transitions as you tune k. And these shape transitions are non-monotonic, as can be described by the non-monotonic behavior of alpha k and gamma k with respect to k. So alpha k is 1 over k plus 2 within this range for k less than 1 but larger than -2, and gamma k is this. Whereas for k greater than 1, the function is different, k by k plus 2, and this is 1 over k. Good. But remember, we are interested in the distribution of the fluctuation of the position of the rightmost particle x max for general k. So once again, of course, on an average, x max will sit at the edge of the density profile, which is Ln, and it will fluctuate around this edge, right? So it will also have some typical fluctuations, and then you'll have some large fluctuation about this mean position. And mean position is into the scales with n as n to the power alpha k. So it turns out to be convenient to scale all the positions in the problem by n to the power alpha k. So that's what we do. We scale all the positions, original positions or xi. We scale them by n to the power alpha k. As a result, we have also scaled the maximum position by n to the power alpha k. And as a result, the density will be within the support of the density will be within -Lk UC2 plus Lk UC, and these are order 1 numbers. So I just wanted to take out all the n factors so that we can see the independence and the explicitly. Okay, so let me give you the result. So for large n, we see that this y max variable fluctuates around this mean value over a scale of n to the power minus eta k. Eta k is another k dependent exponent, and this is the typical fluctuation. And you have a distribution for this, but that distribution does not describe large deviations. Large deviations are actually order 1 distance away from this. It's not of this scale. It's very large from this mean. And they are also described by appropriate large deviation functions, which are k dependent. And the speed here on the left side, this is the left side, is minus sign always corresponds to the left side, is n to the power 2 alpha k plus 1, and on the right side is n to the power 2 alpha k. So it seems that there is also a possibility of phase transition. We'll see that. So let me first talk about the typical part. So in the typical part, if we compute this distribution numerically as a function of y max for different k, and for each k for different values of n, and these are the typical distributions for different values of n. It looks like, and if you plot in the log scale, log linear, this side log and this side is linear, then it looks like this. But interestingly, we see that if we plot this distribution in this form, we multiply y-axis by the standard deviation of y max, which is defined by this, and the x-axis we shift by the mean and then divided by the standard deviation. Then all these curves actually fall on top of each other, and you get a nice scaling data collapse, and that leads to the existence of a scaling function, that the fluctuations of whatever on our scale. So the fluctuation sigma is n to the power minus eta k. As I said here, that this is n to the power minus eta k. So if I write down this as w minus l to the power uc divided by n to the power minus eta k, that is another length scale. So over that length scale, this function is valid, and this function describes the distribution of the typical fluctuation over this. And I should mention that this function is known only for k equal to 0 and k equal to -1. For k equal to 0, we know, as I already told you, it's a Tessier distribution. For k equal to -1, it can be shown that it is a solution of a non-local eigenvalue equation, which already tells you that the Tessier distribution, if you change the interaction, is not. One second, how do I see the question? Yeah, okay, sorry, it is not for me. Okay, so already says that the Tessier distribution is not universal against the change of the nature of the interaction. Now let me tell you about how does this sigma max scale with n. I already told you that it should scale as n to the minus eta k, and let me give you a proof. So we computed this sigma y max numerically as a function of n and for different values of k, and plotted them on log scale, and we see that all of them actually fits very well with a straight line, and the slope of the straight line should give me this, because if you plot log of that, both sides take log, so it should be log sigma max equal to minus eta k log n. So in the log sigma max versus log n scale, it should be a straight line, and the slope will be the exponent, and from this slope we get the exponents, and we plot this exponent as a function of k, and this is the behavior. It's again non-monotonic. The value of the exponents are known explicitly for only 3 values of k equal to -1, 0, and 2. But for other values of k, you have this behavior, and we have some theoretical understanding, some intuitive understanding, but from some other arguments and theories. But I do not have time to go into the details of that. Okay, so I was talking about now I was talking about the typical part. Now let me talk about the large deviations, which are actually largely deviated from the mean, which are of order 1. Order 1 in the scale variable. So in the original variable, it's order n to the power alpha k. So it's very large. Okay, good. So as I told you, that those are described by these large division functions, and we have been able to compute these large division functions, and this is the procedure. This is a bit technical slide. Let me skip it to how to compute these large division functions. But we could compute and find exact explicit expressions of the large division functions, which can be classified into these 3 regimes: -2 to -1, -1 to 1, and k greater than 1. And we could find out that explicit expressions. I just wanted to flash these expressions. These are, I don't expect you to look at them carefully, because they are not illuminating. But I just wanted to show you that there exists explicit expressions for these large division functions. But what important feature that I can bring out from these expressions is that the behavior of this large division function, as you approach this mean value from this side or that side, they again has a power law behavior with exponents that depend on k. And for k greater than 1, this exponent is 2 plus 1 over k, and e plus is 1. And for k greater than less than 1, e plus is 3 minus k by 2, whereas this e minus is 3. And since this speed is different on the left side and the right side, once again, if you define a free energy, as I explained in the context of log gas, that is k going to 0 limit, you see that here also you'll have a function, that is actually the derivative is discontinuous at third order. So the third order transition remains valid for all k greater than -2, even if you change the interaction. And that leads to the fact that the third order transition is actually universal, and it goes beyond the regime of random matrix theory or the log gas. It's actually more universal than that regime. Okay, so these are some plots of the large division functions. The circles are from numerics, and this is the theory. And I am plotting only the function. So the probability is exponential n to the power something times this function. And if you estimate this probability at 10 to the -60, even though we have all plotted only till this much, but we could generate probabilities of 10 to the -100 for this k greater than 1. Here also we could generate probabilities of order 10 to the -100 for this range of k. And for this range of k, we could generate probabilities of order 10 to the -600, even though we have plotted till here, which is of 10 to the -200. And of course, these probabilities we of course could not generate using the normal sampling method. So we use some biasing techniques, which is common biasing techniques, may not be new to this audience. So the method is we have this. So how do you compute? So this is the Maxwell-Boltzmann distribution, and this is the way you can find out the probability of a configuration. And given the probability of a configuration, if you do this summation, you should get this distribution. But we want this distribution at order 10 to the less than 10 to the -15, or in fact of the order of 10 to the -100. So to get this thing, we cannot generate these events. I mean, to get this order of probability, we have to generate these events at least of 10 to the 100 times, which is exorbitantly large. So we have to bias the method appropriately. So to generate Maxwell-Boltzmann distribution in the usual sampling method, usual Markov-Chain Monte Carlo dynamics, you consider a jump dynamics, where you consider start with a configuration and you go to another configuration with a specified jump probability, which is the usual Monte Carlo dynamics probability, jump probability, a minimum of 1 minus the ratio of these two Maxwell-Boltzmann distributions. But as I said, with this probability, with this method, we can only generate probability at minimum of 10 to the -10 probability of order that. But if I want to generate probability of order 10 to the -100, we need to bias, and the biasing is actually done by adding some chemical potential terms to the energy, which actually effectively turns out to be an adding and chemical potential terms, or where theta is an extra parameter, chemical potential. And that's how you can generate. By these dynamics, actually you can actually shift the typical behavior from wherever the desired location. And suppose you want your typical behavior to be at some large value of W, then you have to choose your theta appropriately, and that's how you can actually explore a very large regime. And that's how we can compute this full distribution. Here you do not have to bias, and and there the trajectories are in this form. And of course, in these trajectories, these are typical fluctuations of the X max. This is no bias. But if I want this typical, the largest, the X max to be much smaller than the typical value, that means this is much smaller than this value, then all the particles has to be pushed to the left, right? And that's why you have to choose a theta in such a way that all the curves, all the trajectories of all the particles are actually pushed to the left, this side very much. And that's how we did it. And on the other hand, this side, the most efficient way to create a very large fluctuation is that only the rightmost guy goes very far to the right, but all the other particles are sitting happily within the normal range that it actually does here without any biasing. And that's what we see in the simulation also, that only the rightmost guy actually goes very far, but all the other particles are sitting here. And the corresponding density looks like this, that you have a density here, which is similar to this, not similar in the larger limit, exactly same, but only one guy which has gone outside of this sea of particles. Whereas here, without any bias, this is the usual density. Here I am giving the schematic plot only for k equal to 0, but you have similar plots for other values of k. This side, the density is looking like this, as if that there is some wall which is pushing all the particles, because that is the optimal configuration that actually creates such a large fluctuation. And we have to find out the probability of such density fluctuation, density change, deviation. And as a result, you see that quite expectedly, you see that the density is actually very large at the edge, is very large because you are pushing, and because of the harmonic potential, the interplay between the harmonic potential and the repulsion, the particle starts accumulating, and there is a large density here. But this side you have a this side is not getting affected, but this side you get a very large density. Okay, so so once you generate a very those events, then how do you compute the probability distribution of the Ymax? So by doing this thing, you can get a distribution, but that is for the bias dynamics. But you have to get it for the unbiased dynamics. But you can see that easily that this distribution is related to the unbiased dynamics by this factor. So by doing this transformation, you can get it. Of course, this equal to sign is not correct. It should be proportional to that. And by choosing one theta, you cannot explore all the full of the full range. So you, for different ranges, you have to choose for different theta and explore different regimes. And for with different theta, you explore different regimes. For example, theta greater than 0, you explore only the left regime. Theta less than 0, you explore only the right regime. And once you get the different portions of the original curve, you have to scale back and glue them, and that's how you get the original unbiased distribution properly. Okay, so so I'm at the end of my talk. So this is the conclusion. So we have studied extreme statistics in a gas of particles in equilibrium which are confined by harmonic potential interacting by long range interaction. We showed that the changing interaction term shows that the Hessian distribution no longer describes the typical fluctuations of X max. However, the large deviation functions still exhibit third order phase transition, extending the universality to models outside the regime of log gas or random matrix theory. And we also described an important sampling method for generating the rare events described by these large deviation functions. So these are the references on which this talk is based on, and these are my collaborations. So these are the students. This is my graduate student who has whatever I have talked mostly is done by this guy. These are the recent results, but there are some supporting results which was done by this was a formal long-term student at ICTS, and this was my research assistant who was visiting at ICTS, and these are my collaborators. Many papers are written with them. And once again, I would like to acknowledge for financial support from CEPIPRA and ACLB Government of India, and thank you.
Yeah, thank you, Anupam. Yeah, for a very interesting talk. So actually, I don't see any question in the chat, but I have two questions to ask, and since I'm not from the same background, the questions might seem naive. So one is that you are talking about large deviations, and the large deviations, the way I understand that it is occurring at the tail of your distribution. So my question was that the normal deviation versus large deviation, that proportion, does it increase or does it change based on the position in the distribution? So that means, is it more in the tail of the distribution or less?
Yeah, of course. If you look at the ratio, the probability is decaying. So if you look at the ratio in which the large deviation is at the denominator, so it will increase exponentially, of course, as you go farther and farther.
Okay.
Because the probability is decaying further and further with exponentially.
Okay.
So the ratio should increase.
Okay. And so another thing that I did not understand is that the K was minus, in the negative side, it was up to -2. So
yeah. Okay. So I did not explain this region because so if you look at the form of the potential, let me go to the form of the potential. Sorry, is the potential? Yeah, this one. If K is actually -2, then it's actually on the xi minus xz square, right?
Mm-hmm.
So these two terms are of comparable strength. Now, if it is less than -2, then it will be xi minus xz to the power 3, let's say. Okay. So this will be so dominating that all the charges will be either at the plus infinity or minus infinity, or depending on this thing. So then you will not get any non-trivial density profiles, non-trivial physics. So that's why you have to consider k. But if you change this power, then also you can change this power. You can limit this power by this.
Got it.
Since here I have considered harmonic potential, that's why you have to consider k to be larger than -2.
So now, this is I think a one-dimensional model that you're talking about. So is it so typically we will talk about jumps and large deviations in glassy systems, supercooled systems. So is there can you tell that how that this can be connected to those kind of systems?
As I said, the other model, this log gas model, where this interaction is logarithmic, there are some connections with spin glass. As I said, if you consider random potential, where the potential is a function of n variables, but they are the values of this potential in one dimension, of course, is chosen from Gaussian distribution. So let's say you have a potential V of x1, x2, xn, n variables, and this potential is chosen from Gaussian distribution. Then if you consider the minima of this potential, the statistics of the minima of this potential, then there is a mathematical theorem called Kac-Rice theorem. But according to this theorem, you can connect this problem to a problem of random matrix in Gaussian unitary ensemble. And then you can show that you can see that the number of minima undergoes a transition that is predicted by this log gas. There is some connection, which is difficult to say in just words, unless I show some expressions or do some. But okay. This is the best I could say in words.
Sure. Okay, so thank you, Anupam, again for an interesting talk. So I think that brings us to the end of this particular session. But I would like to tell the audience to go and please visit. There are very interesting posters there. Whenever you have time, please go and visit those posters. The link for getting to the posters is given in this chat box, and also probably already you know about it from the emails and all. I think you have to sign up first before getting into that. So with that, I will hand over to the organizers, Nishant and Jagannath. So.
Thank you, Arnab. Jagannath, you can announce the posters.
No, I think so. Basically, I think what I think that Arnab tried to point out is that so tonight, at least in Indian time, and in the morning, I think in United States time tomorrow, I think we have the formal poster session. But considering the time zone, I think you can join anytime. And you can at least look at all the posters and place your comment there. And it will be really, appreciated if all of you take the time to at least come for that poster session and basically interact with all our poster participants. So those posters that we basically have, I will say that many of the posters are excellent, and many of the people which you could not get for seminars, their group members are presenting. So definitely it will be really a treat if you can actually have a time to look at it.
Okay, so thank you everyone.
Yes.
Videos are posted on YouTube channel, so you'll be able to see that.
Bye. Nishant,
yes.
Nishant, this is Shravani here. So
yes.
In the next session, will you also talk about the Google Doc that you have posted? So yes. People can leave their thoughts over there so that we can just mention it in case they haven't seen the email.
Yeah, we will start with that, right, Shravani? We will in the beginning of the beginning of the session. Yeah, sure.
Okay, thank you.
Yeah, thank you. Bye.
Bye.